pays ni leurs institutions. Donc, sans EPC, on a pris le package suprême, le paquet diamant. C'est une revue et je suis contente que le débat sur les langues ait déjà été posé parce que c'est un débat qui est politique, c'est une question d'engagement et de volonté. Jean l'a dit, c'est une revue qui publie en anglais, en français, mais aussi dans les langues africaines. L'arabe, on a commencé par le swahili, mais nous espérons au moins que les abstracts vont être maintenant progressivement traduit en Hausa, en Bambara, en Fulani, en Yoruba, en Igbo, etc. Et ça dépend vraiment de la volonté des chercheurs. Nous sommes en train d'essayer de voir ça. C'est quelque chose qui est politique et qui est extrêmement important et personne ne le fera à notre place. C'est extrêmement important. Donc, tous les articles, et ça c'est très important, tous les articles sont traduits minimalement en français et en anglais. Et tous les articles ont leurs résumés qui sont traduits dans les 4 langues, nécessairement. Ça demande beaucoup de temps, beaucoup de ressources. Et à la suite de Jean, je demande aux bailleurs dans la salle de nous appuyer dans cette politique des langues parce que ça prend du temps et ça prend beaucoup de ressources. Mais il faut le faire. Nous sommes aussi une revue trimestrielle, donc nous publions 4 numéros par an parce qu'il y a énormément de possibilités et de capacités. Et vous pouvez vous dire, mais pourquoi une revue en sciences humaines et sociales se trouve ici alors que, voilà. En fait, et je vais finir très rapidement, monsieur le modérateur, nous travaillons sur les transitions, nous travaillons sur les enjeux économiques, énergétiques, démographiques, numériques, sanitaires, écologiques, technologiques, etc., à partir d'approches pluridisciplinaires et aussi interdisciplinaires. Je crois que nous sommes tous conscients que le silo disciplinaire est quelque chose qui ne fonctionne plus et nous devons apprendre à faire de l'inter et de la transdisciplinarité. Et j'en veux pour exemple le numéro 2 qu'on a publié au mois de décembre, Africa and the World in Viral Times, l'Afrique et le monde durant la Covid-19, et vous avez ici le lien. Et dans ce numéro, qui était absolument passionnant, je trouve, nous avons évidemment une masse importante de socio-anthropologues, mais nous avions aussi des épidémiologistes, dont le directeur adjoint de l'OMS, qui est aujourd'hui en charge des maladies tropicales négligées, Ibrahima Socéphal, qui a fait une interview, ainsi que le directeur du Centre de recherche et de formation en infectiologie de Guinée, le CERFIG, Abdoulaye Touré. Donc, des épidémiologistes ont publié dans ce numéro, des climatologues. Hier, il y avait une discussion extrêmement intéressante sur pourquoi l'Afrique a eu moins de cas. Ils ont éliminé la question climatique. On a beaucoup dit que c'est parce qu'il faisait plus chaud ici. Ils ont prouvé que c'était vraiment quelque chose à discuter, à revoir. Nous avons des écologues qui publient. Les revues sont vraiment ouvertes. La revue est ouverte aux jeunes chercheurs, aux chercheurs juniors. Nous avons 2 doctorants qui ont publié dans ce numéro, à côté de grands noms dans le monde des sciences humaines et sociales. On connaît tous Didier Fassin, Collège de France, etc., extrêmement reconnu, a publié à côté de jeunes chercheurs. Ça, c'est aussi un équilibre très important à trouver. Et le constat général de ce numéro, entre autres choses, c'est qu'il n'y a pas de revue francophone africaine en santé publique. Et ça, c'est quelque chose qui est extrêmement important. C'est peut-être aussi ma deuxième interpellation à WANIDA, mais pas seulement WANIDA, l'ensemble des réseaux de ACE Partners sur l'importance, dans la construction en amont des projets, de mettre des éditeurs. L'édition, ce n'est pas quelque chose qu'on pense quand on arrive, quand on a des résultats. C'est quelque chose qu'on pense en montant le projet pour accompagner, regarder, etc. Et c'est ce qui évite que, même si c'est super, que vous publiez tous exclusivement dans des revues qui sont basées au Nord. C'est extrêmement important. Les éditeurs doivent être en amont. Et donc, oui, j'ai juste mis deux superbes citations, je pense, d'Ibrahima Socéphal, encore une fois, qui explique, et ainsi qu'Abdoulaye Touré, que maintenant, pour tout ce qui est épidémiologie de terrain, les meilleurs chercheurs sont aujourd'hui en Afrique. Que chaque année, on a plus d'une centaine d'épidémies sur le terrain et il y a une expertise continentale qui est de plus en plus fascinante et féconde et stimulante. Et c'est important que la publication de ces résultats ne se fasse pas uniquement, même si je n'ai rien contre, de Lancet Science, etc., mais aussi dans des revues qui sont portées ici sur le continent. Ça fait aussi partie disons, du travail à faire. Je vous remercie.
Merci beaucoup. Merci. Merci, professeur Ba, pour cette excellente présentation qui met le doigt sur un autre aspect de la valorisation des travaux de recherche qui sont conduits en Afrique. Donc, faciliter l'accès au plus grand nombre, c'est aussi un environnement dans lequel il faut innover. Et cette présentation, elle met le doigt, et je pense qu'il y aura beaucoup de questions, beaucoup d'échanges, beaucoup d'interactions pour voir comment ensemble on peut aborder la question et contribuer. Donc, je pense que des questions sont probablement là, mais je ne vais peut-être pas prendre une question maintenant. On va continuer tout à l'heure. Merci encore. Alors, je vais introduire maintenant le dernier orateur de cette session. C'est le docteur Onan Jambou qui va nous présenter le CERMES du Niger, donc qui fait partie du réseau des Instituts Pasteur, c'est le centre de recherche médicale et sanitaire. Donc nous accueillons maintenant nous donnons la parole au professeur Onan Jambou pour nous présenter ce laboratoire qui vient de rejoindre le réseau WANIDA avec l'IRCV du Niger.
Okay, thank you, Professor Nikkei. So I will follow Professor Yap. As we are in Ghana, I will speak in English, even if I totally support the previous talk of my colleague about multilanguages. So it's a pleasure and an honor to be here and to join the WANIDA network. I will speak, I'm the scientific director of CERMES, and I will speak on behalf of my general director, Professor Awasabo, who is currently in Paris, so we switch the destination. So CERMES is a public institution in Niger. Yes. It was created in 1980 as on behalf of the West African Organization for Disease Control, and after several years he joined the Pasteur Network. Now it's a public institution from the Health Ministry, but affiliated to the Pasteur Network. So, as I said, our boss is the Ministry of Health, of course, but we have also formal linked with the Ministry of Higher Education and Research and Finance, of course, but also with the Pasteur Network, because we have a convention with the Pasteur Network and the Institut Pasteur in Paris. And we have also informal link with the university, regional health institution, and regional academic institution. So we have a lot of partners to support our research and actions. So nowadays, the vision that we have is about to anticipate the problem of health in Niger. To anticipate the problem of health in Sahel for the 10 next years, because the context is changing rapidly, especially in climate and urbanization and so on, and so we have to anticipate. So we have the 4 traditional Pasteur mission, that mean research, public health, training, expertise, and we are also the organization, National Institution of Coordination in response to epidemics in Niger. We have several agreements and conventions with university, with colleagues from LASDEL, and also with a university in France and in US. And we host 8 national reference laboratories, as we are a public institution, and we are also in charge of response to outbreak, and especially also on malaria control, and coordination with the regional institution of health. So to do that now, we are a small institution, about 100 staff with an average of 20 students. And we have 4 units: bacteriology, virology, epidemiology, malaria and entomology, and also platform that we settled recently, especially immunohematology, and the chief of the platform is with me. It's Ibrahim Karidio. And we have a quality insurance services. So the topic of the CERMES changed along the years, of course, and it started with meningitis, schistosomiasis, and now we move to respiratory disease, polio, emerging disease, malaria, and of course, COVID. So we have a training center. Training center has a great role for the training of healthcare workers from the public institution, also for students. So we have training modules in public health, intensive course, master modules. And now we are setting e-learning platform to disseminate the modules to all the country, because the country is very large and it's quite difficult to have all the students from everywhere. And also the training centers is in charge of the communication. So the current activity that we have in CERMES. First of all, for the last 2 years, of course, we were working on COVID. In epidemiology, mainly on healthcare workers and their family, blood bank, delivery women, general population, and also on variant of the SARS-CoV-2, especially in virology, also genetics diversity studies, and diagnostic tests. The study, but also we have to settle, most of all, to settle the diagnostic for all the Niger, because there are no other health research center in the country. So we were in charge to settle the diagnostic for all the Niger, and after for the travelers. So we have to manage 12,000 tests a month for the last 3 years. Other topics in CERMES: meningitis, healthcare of women, respiratory viruses, survey of hemorrhagic fever, and vector and plasmodia distribution and resistance, and study on malaria control, adaptation of the strategy with the national program. And also in bacteriology, survey of several bacteria and epidemics, and also program with colleagues from the US on mass treatment with azithromycin. And the new platform of immunology is now working also on a disease from the red cell, that means sickle cell anemia, thalassemia. And also we are developing now a program on cervical cancer and hepatocarcinoma. So where are we going for the next 10 years? Oh, something happened. Something lost. So if we are looking at the map, where is the Niger? It's simple. We are in the center of the Sahel area. So we have to work on Sahel and on changes that will occur in the Sahel. And it's why we discuss a lot the last 2 years about what will be the future of CERMES for the next 10 years. And we discuss about determinant in change of the Sahel, and we list them. And we have very important change that will occur, especially climatic change, but also urbanization, migration, because you know that Niger is a center point of migration from south to north and to in the African region, West African and Central African, legal or illegal migration, of course, but it's this migration have a very big implication in public health. So the major threat that we decided to address was most of all women health, of course, malnutrition, but emerging disease, and also drug resistance and newly emergence of cancer in relation with virus, environment, and so on. For that, the capacity that we need to develop in CERMES are modeling, statistics, technical platform, genomic approaches, and we already started with that thanks to COVID, and social science and remote sensing to address questions about the the spread of disease according to all the changes in Sahel. And so we need to improve and to increase human resources through network like the WANIDA, through convention with university and institution, and also with all the colleagues in the north. So we decided to focus on some transversal axes, mainly mother and child health, malnutrition, climate change in the Sahel, climate change or the urbanization, and disease propagation in this area, and epidemics. And for public health, we have to organize a network in the country, but also network with colleagues in the different countries of the Sahel to organize survey and to start modeling propagation of disease in this area according to all the changes, and especially climate changes. And so, of course, we will continue training and for healthcare workers, but also we are now organizing a summer school of nutrition with the University of Grenoble, and also we are starting to organize for this year new courses on genomic survey of meningitis. And we have also scientific days every 2 years, so it will be this year's, and we will happy to welcome all of you in December, first week of December, for the 4th scientific days of the CERMES. Thank you very much.
Thank you, Ronan. I think we still have time to take 1 or 2 questions before I give the microphone to our host, Professor Wandari, to close the session. If there is no, I can ask one. Hold on. Now that you are a new member to this network, you want a comment and your expectation. What do you expect from this? How do you think the network can benefit SEMERS?
The main thing that we can organize is to first to apply for grants altogether, to exchange student and researcher, especially to exchange our technology, to use our technology platforms, and not to go each time to the north. It's better to stay here. I think that we have a lot of opportunities altogether for training and for also for platform. Maybe we can also think about going together to buy equipments, because each time we are several institutes, we can obtain better prices, especially for genomics. If we want all to buy a sequencer, so maybe we can apply all together to buy 10 and not 1. We will have better prices. And also for maintenance. The major problem that we have in all the country is maintenance of the equipment. Maybe we can thinking to organize a network of maintenance, or maybe to apply for contract with the firms, but all together, because it will be cheaper to organize that. I think that maybe that seems to be housekeeping activities, but we have all this problem. To get reagents and to have maintenance in Niger, it's quite not easy. So I think it's the same for everybody in this room. So we have a lot of things to discuss, sometimes practical things. Not maybe high science, but practical things also, because it will be efficient, I think.
Thank you, Renan. I think this is also a very important point, how WANIDA can be a negotiator for services and supplies on the continent. I think this is one of the major points. I will let Gordon now conclude this session, and I also want to thank Professor Renan for this wonderful talk, and ask for this applause.
Good morning. I didn't hear a response. Good morning. I don't think anybody's hungry, no? I think it's been an exciting morning. Basically a demonstration of how much opportunity there is for us to work together, and the potential for a lot of significant change and impact if we actually talk to each other and look for synergies in what we're doing. All of us sitting here from so many different African countries, we have common challenges, we have common visions. We are all striving to achieve the same thing, which is an Africa that is independent and that is capable of finding solutions to our own challenges, obviously with the support of our global partners. But this is a common vision that we all share. And I think that in order to achieve this vision, we need to work together. Of course, there are challenges like all the speakers have mentioned. Even traveling to come here, there were challenges. The other day we were talking during the break about how easy it is to go to London. Much easier for me to go to London than to go to Cameroon. Not to even talk about Niger. Okay. Sometimes I feel like it's better for me to just drive and go than to try to do the bus the plane stops that are like bus stops. You take the flight and they take you 4 countries forward and bring you back 2 countries, or you go west, go east and come back to Central Africa. So there are some challenges, but that's why we are a resilient continent. Africa has faced so many challenges, but we always survive and make progress, and we shall do so again in terms of research. And this meeting is a very positive step towards making such progress. WANIDA has provided a solid platform for us to network and to work together. And I've been so impressed by this meeting, the level of engagement and participation and enthusiasm from everybody to try to engage and make it count, make the meeting count, and build partnerships. I've seen a lot of side meetings taking place, and that is the spirit on which this conference was organized. So we would end the session now, take a break, continue more discussions during the poster session, and then we'll reconvene at 11 or so. What time is it?
I think, we'll now go for break, and I think there are some indications that the
Some
moderator will
announcements?
tell us. Yeah.
Okay, all right. So
Thank you, Gordon.
thank you very much.
Yeah.
Good. Can I have everyone come back to the auditorium, please? Can we all please be seated for us to start the next session? Our next presenter is online and ready to present, please. No.
You just keep and go through
Yeah, so I mean, I'm just going to call the
chair.
The chair first, and then yeah. Right. So before we call the chair to come in, we all know that there's a breakout session and there's a room at the other side, room 2. Unfortunately, we have only 4 people in that room. So please, let's go and support our students. So we have Mr. Podo, we have Christine Moore, and we have John Sandy, who are both presenting at the other side. So please go and give them all your support. So let's fill the rooms, please, and not be all, on one side. Otherwise, it will be imbalanced. Right. So our next session for the time being is still under the theme that we had earlier, and our chair is in the person of Dr. Aliu. So Dr. Aliu, if you're around, could you please take over from me and then chair the session? Thank you. And please be widely awake whilst we listen to the presentations. Thank you.
Thank you, Gloria. Thank you. I want to appreciate WANIDA for organizing this very memorable occasion. It's obviously a very huge success. Our next session will start with an online presentation, but it seems the presenter, is the presenter around? Okay, the online presentation has been shifted, so I want to see if the Professor Amidou Sami is around. Professor Amidou Sami is an associate professor in the Department of Microbiology, University of Banda, South Africa. He's going to present on interleukin-10 production and seroprevalence of entamoeba histolytica infection among HIV-infected patient in South Africa. Professor Amidou, welcome.
Yeah, good afternoon, ladies and gentlemen. Welcome to this session. I would like to continue with, by saying congratulations to the WANIDA team for their brilliant organization, and also to say thank you for inviting us and giving us the opportunity, to share and to be able to create new links that might lead to further development for the young scientists in the continent. So I'm going to present a piece of work that was done by one of my PhD students. Dr. Ngobeni, who is now a postdoc in the US, on interleukin-10 production and seroprevalence of entamoeba histolytica. So as a guide of introduction, diarrheal diseases, as you know, is a very important cause of morbidity and mortality, particularly among children in many developing countries. And one of the causes of diarrheal diseases is entamoeba histolytica, which causes amoebiasis. This topic is very special because almost nobody has done studies on entamoeba histolytica since we started the symposium, and it is I believe it's an important cause of mortality in many countries. So if you look at the graph, you will see that diarrheal diseases represent 13%. 13% of all deaths, among children, followed by, AIDS, pneumonia, and then others, which are responsible for mortality among children less than 5 years old. And amoebiasis is the infection by entamoeba histolytica, and it's one of the most problematic because it has two different types of disease presentation, the intestinal and extraintestinal. Although the extraintestinal infections only constitute less than 1%. So this is the life cycle of amoebiasis. So generally, either we can get the cyst from food, from our hands if they are dirty, or from water that we are drinking. So we get the cyst, and then those cysts are ingested. So once in the stomach, then they are excisted, and then in the intestine, then they are going to cause the disease. They can cause colitis, and then some of the entamoeba trophozoites are going to become cysts, and then will be excreted, with the stool samples. And those sometimes can find their ways into water systems, or can contaminate vegetables, or can be also transported by flies to the food that we are eating, and then we get contaminated again. So once entamoeba histolytica get into the stomach, so then they are going to produce enzymes, particularly cysteine proteinases, as well as many other enzymes that are going to participate in the degradation of the mucous layer on the intestine and facilitate the infection by entamoeba histolytica, which is going to produce those more cysteine that are going to degrade the wall, and then there will be activation of a certain number of cytokines, and we can see, for example, IL-1 beta, IL-1-6, IL-8, IL-12, interferon gamma, TNF alpha, and these the product of these cytokines are going to create the inflammation in the intestinal wall and then lead to colitis. So invasive diseases in the case of entamoeba histolytica infections include liver abscess, which is the most common, approximately 1% of symptomatic cases. Although in some countries, like in Japan, the percentage has reached 17%. And I think that occasionally in some European countries, you might also have a liver abscess, which is much higher in some cases, even without the intestinal infections. Astonishingly, very little is known about the immune response underlying the invasive amoebiasis. And previous studies have shown that interleukin-6 was found to have significant levels in some patients that has amoebic liver abscess. Although few studies have also indicated the role of interleukin-10 on the protection of the host against entamoeba histolytica, but those studies were conducted in mice, and no study has been conducted in humans. So the objectives of the study that we conducted were to determine the seroprevalence of entamoeba histolytica, to estimate the occurrence of extraintestinal amoebiasis by the detection of antigen in the circulating blood. Then we also determined the impact of interleukin-10 on the occurrence of IgG among asymptomatic HIV-positive patients. So material and method. So this is our study site. Our study site is situated in the northern part of the country. This is South Africa. Cape Town, most people when they go to South Africa, they visit Cape Town, but we are far in the north. We are somewhere around here. This is Polokwane, and then Louis Trichardt and Tohoyando is somewhere around here. So this is the Vembe district where the samples were collected, but most of the samples were collected in Polokwane, which is the capital city of Limpopo province. We have Pretoria here, and then Johannesburg down here. So what we did, we collected blood samples from asymptomatic HIV patients. So generally, these are the patients that come to the hospital for a visit, either for their CD4 count or to check the viral load or to collect their medicine. So these patients, they were asymptomatic. They did not have any symptoms. So we use a questionnaire in order to be able to collect some information, like demographic information in terms of origin, age, sex, as well as the CD4 count, and also to know if the patients had other diseases like STDs. So we use the TapeLab 2, the last entamoeba histolytica test 2, antibody detection, and also the entamoeba histolytica antigen detection. So both these two kits, they came from TapeLab from Virginia. Sorry. Then we use another kit, ELISA kit, in order to be able to determine the concentration of interleukin-10 among these patients, and the results were analyzed using SPSS. So in terms of the results, this table shows the demographic characteristics of the study participants in terms of the origin, the district where they came from, the area, whether it was rural or urban. We also look at whether the sample was obtained from the hospital, the clinics, as well as the gender, the age group, and the CD4 count level. So the seroprevalence of entamoeba histolytica was found to be much higher from the hospitals, from patients who were recruited from the hospitals, and the p-value was significant only at that level. In terms of gender, there was no significant difference, and also for the age group. So it did not matter what the age the patient was, but the prevalence of the seroprevalence of entamoeba histolytica was quite constant among these different groups. So the samples, when we tested all the 364 samples, only 3 samples were positive for the antigen. So we wanted to look at those samples to see what were the characteristics. So the age varied from 29 to 64, and all the 3 patients were females. And then they came from 2 different districts, and the area where they lived was mostly rural. Their CD4 count was above 200, which is about 350 on average. And in terms of the antibodies, 2 of those samples were positive, while 1 was negative. So in terms of the entamoeba antigen, so they were positive. And for the IL-8, all the 3 of them had a lower concentration of interleukin-10. So in terms of the distribution of IL-10 by age and by gender, so there was no significant difference among the different age group and gender. So in terms of the occurrence of entamoeba histolytica in relation to the concentration of IL-10, So we can see that those samples that had higher concentration of IL-10, they had much lower prevalence of antibodies, and the p-value was significant, 0.006, which is less than 0.05 that we're targeting. So this is an indication that patients that had produced more IL-10, they were kind of less as risk compared to those that produce less IL-10. So these figures also show similar results. So we can see on the figure on the left, we have the participants with IL-10 more than 2,000 picogram. And then on the figure on the right, we have entamoeba histolytica seroprevalence. So we can see that those patients that had lower CD4 count, they had more infections compared to those that has lower CD4, that has lower IL-10 concentrations in their blood. So in terms of the conclusions, we demonstrated a high seroprevalence of entamoeba, which shows that this infection is quite common in the area, with about 65% of individuals living in the area having been in contact with the pathogen. Amoebic liver abscesses might be less common because of the prevalence of 0.5%. Elevated levels of IL-10 in patients may serve as protection against the parasite. So we were able to show that the higher the level of IL-10 in the serum, the lower the seropositivity. So the occurrence of entamoeba histolytica in this study is of public health significance, and if appropriate care is not taken, it could result in epidemic situations. So this study also observed that the infection is more prevalent in rural areas. If you look at other studies in other countries, like in Bangladesh, for example, the prevalence of entamoeba was much higher in townships, compared to the villages. But in this case, we found higher infections in the villages, in the rural areas, compared to town. So I would like to thank our funders, the National Research Foundation of South Africa and the Department of Science and Technology, and the patients who participated in the study, and the students who helped with sample collection. Thank you very much.
Okay, thank you very much. So we open the floor for questions, comments, or contributions. Okay, there is one.
Was there any correlation between the amount of IL-10 and the amount of interferon? Did you measure the pro-inflammatory cytokines?
No, we did not measure the other cytokines. So because we went from the hypothesis that was generated by Hamano and his colleague in 2006 at the University of Virginia, who demonstrated using the mice that mice that had high IL-10 tend to be protected. So we wanted to verify that in the humans. So we did not test for other cytokines.
Okay, because IL-10 is often the end product of an inflammatory response, like interferon, TNF, and so you get an inflammatory response in your liver abscess, which then induces IL-10, rather than it's actually protective.
Okay.
I mean, it might be worth looking at the other cytokines to see over time as well how it changes.
Okay, yeah, thank you for the advice.
Any other question?
Yeah, hello. Thank you for a great talk. I just wanted to know what influenced the cutoff for your IL-10 concentrations you used in your study?
Yeah, it was based on the overall distribution. So we tried to find a middle way. So it was not informed by any official data. So we just decided based on the overall distribution of L10 in the population. So then we wanted to characterize them, one group that we considered lower and one that we considered higher. Yeah.
Yeah, I'm just asking that because you base on this cutoff to tell this is high and this is low. And then at least based on what the earlier question that was asked, at least if you were able to look at the ratio of this L10 to any pro-inflammatory cytokine response, that could have been more informative.
Yes.
Yeah.
Yeah.
Okay. Any other one? So in the absence of any other questions, thank you for the presentation. I'd like to call on the Dr. Firman Kara. Dr. Firman Kara, who is a social anthropologist, Institute of Robert Koch, Chair UNESCO, the Biotechnique University, Alessandra, Côte d'Ivoire, who is presenting on the preparedness to response, a social anthropological contributions from study of the funeral rites in Côte d'Ivoire. Thank you. You're welcome.
Okay. Bonjour à tous et à toutes. Alors, voilà. Depuis 3 jours, nous avons écouté les sciences biomédicales. Et ce matin, je vais vous parler des questions culturelles et sociales. Donc, ma communication est intitulée de la préparation à la réponse, une contribution socio-anthropologique à partir d'une étude de dérive funéraire en Côte d'Ivoire. Donc c'est pratiquement une dimension appliquée de la communication que j'ai faite le premier jour. Donc, il faut dire que ce travail a commencé en 2015 lors de l'épidémie de la maladie à virus Ebola en Afrique de l'Ouest. Et pendant cette épidémie, nous avons mené une recherche qui portait sur 5 pays de l'Afrique de l'Ouest. Et dans ce programme en Côte d'Ivoire, j'ai fait mon master. Et pendant cette période, mon travail de master a consisté à faire une analyse du vécu social des mesures qui interdisaient la manipulation des corps en Côte d'Ivoire. Et quand on a fini cette recherche le 15 mars de 2015, on est allé faire la restitution. Et à la fin, le directeur de l'NHP, qui est une institution qui est en charge de la mise en œuvre des politiques de réponse en Côte d'Ivoire, a pris la parole et il a dit: si vous devez continuer les recherches sur une seule chose, ça sera sur les rites funéraires. Donc c'est à la suite de ça qu'on a mis en place le programme rituel funéraire et épidémie en Côte d'Ivoire. Et on a mené les recherches parce que cette question, on l'avait en Guinée et au Congo, a été parfois très complexe et délicate. Donc le type de mon travail, c'est de montrer comment la socio-anthropologie peut contribuer à articuler impératifs culturels et non biosécurité. Donc pour faire ce travail, nous avons donc décidé de faire une ethnographie des dérives funéraires donc pour se préparer. Donc on est parti des méthodes classiques de l'anthropologie, telles que l'immersion longue et la recension de documents avec des entretiens approfondis. Donc notre travail a consisté donc à faire une anthropologie des occasions de contact, qui a consisté donc à identifier sur la base du risque construit par la santé publique. Nous avons essayé d'identifier les moments où on touche le corps et les moments où on ne touche pas le corps, et les logiques qui font qu'on touchait ces corps-là. Donc à chaque niveau, nous avons recherché donc ce qui est interdit, ce qui est obligatoire et ce qui est permis dans ces pratiques. Et ce travail a permis donc de prendre la mesure de ce que peut induire l'introduction des normes biosécuritaires, et ensuite d'identifier les contre-rites et les fenêtres d'opportunité pour essayer de comprendre comment adapter les mesures de biosécurité à ces pratiques. Et à l'issue de ce travail, il faut, nous avons tiré quelques leçons. C'est que pour les populations, tout cadavre comporte un risque. Donc ça veut dire que avant même que la santé public n'a communiqué sur les corps comme étant des objets à risque, et ils le savent déjà. Ensuite, nous avons identifié aussi que la dignité, la question de la dignité varie en fait selon les aires culturelles, et aussi selon les situations de décès. Et aussi, c'est que cette autre question des dérives funéraires est imbriquée en fait à des logiques de réputation. Nous avons aussi trouvé que toutes les situations de décès font appel à des arrangements dans les enterrements. Et ça veut dire qu'on peut aussi utiliser ces pratiques-là pour adapter les mesures. Et aussi, nous avons trouvé que la variabilité et la diversité des rites funéraires donnent des opportunités d'adaptation, contrairement à l'idée de la complexité. Et aussi, on a trouvé que la méthode d'immersion longue ne peut pas toujours être appliquée en situation de crise. Donc, nous avons proposé que pour répondre aux épidémies, il faut déjà considérer les risques funéraires dès les premiers signes administratifs. C'est-à-dire qu'il ne faut pas, comme on a pour habitude de communiquer pour dire que de ne pas manipuler les corps, mais les risques funéraires, c'est pas après le décès, mais ça commence dès les premiers signes de la mort. Et aussi, il faut promouvoir une approche systémique intégrée, une approche de la construction. Et il faut aussi réviser les méthodes anthropologiques pour les adapter. Et c'est cette base, à partir de ce que nous avons appris en Côte d'Ivoire, En 2020, j'arrive en France et c'était en pleine période de la première vague de la pandémie. Et l'occasion m'a été offerte d'intégrer un programme de recherche à l'hôpital européen de Marseille. En mars, le 30 mars 2020, j'assiste à leur première réunion de crise, et ce jour, la question qui était à l'ordre du jour, c'était la question des rites funéraires. Ils avaient une grande préoccupation parce qu'ils ne savaient, c'était interdit et ils ne savaient pas comment prendre en charge cette question. Et donc, je procède à une analyse contextuelle rapide de l'environnement même de l'hôpital. Il se trouve que c'est un hôpital qui est fréquenté par un groupe assez hétérogène, donc les Maghrébins, les Gitans en particulier. Ensuite, les relations sont parfois tendues, d'où la peur des médecins par rapport à cette question en cette période de crise. Donc, la question des délits funéraires, on voit que c'est transversal parce que quand la question s'est posée, comme partout dans les hôpitaux en France, l'hôpital européen de Marseille va sectoriser ces différents services. Donc, le service, il va avoir service Covid et service non Covid. Ensuite, l'équipe mobile va être divisé en deux, et il y a une partie qui garde le nom de l'équipe mobile des soins palliatifs, et l'autre va être dédié au service Covid. Et le problème, c'est que tous ces services qui étaient divisés étaient restés ouverts à la morgue, sauf la morgue avait accès. Donc, on a échangé, et les premières discussions ont permis de savoir que pour pouvoir prendre en charge cette question, il faut recueillir les informations et les transmettre aux responsables de la morgue afin qu'ils puissent anticiper l'accueil des familles. Pardonnez. Ensuite, nous avons identifié qu'il fallait donc il fallait aussi mettre en place un dispositif de travail collaboratif, de sorte à mutualiser les savoirs, parce que habituellement les équipes ils ne travaillent pas avec le responsable de la morgue. Et aussi, on a trouvé qu'il fallait aussi trouver un moyen pour maintenir le lien entre les équipes de soins palliatifs et la suite des événements mortuaires. Donc, suite à ce travail de choses d'analyse contextuelle, nous avons proposé de construire une cellule rite funéraire au sein de l'hôpital, et qui avait pour objectif de encourager le zone.
Can you round off in the next 2 minutes? 2 minutes. Round off.
Donc, de fini. Ensuite, cette cellule avait pour objectif donc de soutenir toute l'équipe pour qu'ils puissent travailler ensemble. Donc ça, c'est le témoignage de du médecin de l'équipe mobile, donc j'ai pris sa phrase pour faire le titre, penser les morts pour penser les funérailles pour penser les morts et préparer les vivants. En gros, c'est que ce travail permet non seulement d'aider les familles, mais aussi de permettre aux équipes de soins de pouvoir se sentir accomplir leur travail. Donc, en conclusion, nous disons que ce travail donc permet donc de rappeler qu'on peut mener une étude au sud, une étude une perspective au sud et aller l'appliquer. Et cette étude qui se fait dans un contexte qui n'est pas nécessairement un contexte d'épidémie, mais les résultats peuvent être appliqués ailleurs, et surtout de faire la promotion de l'approche de la co-construction, parce que les décisions doivent être prises en commun accord avec les familles des personnes malades, puisque c'est eux qui savent, qui connaissent l'essence de leur pratique et de ce qui peut être digne pour les morts. Je vous remercie.
Okay, thank you very much, Dr. Furman. Before I open the floor, obviously it's an interesting topic, looking at the cultural diversity of Africa and how serious we took issue of parental right. How do you think a sociologist will work with the biomedical scientist to address the issue of belief we have that we have to take our dead bodies, we have to give them the rights they have, and then not giving them is taboo in some communities. So how do you convince people that in some certain situations such kind of things might be dangerous to them, looking at the risk in handling especially dead bodies from the pandemic? Thank you.
C'est comment j'ai pensé le risque et
how do you convince let's say traditional rulers, community leaders of knowing the risk, the biosafety risk in taking dead bodies of people who died of infectious disease, especially during pandemic, Ebola, COVID-19, and so on and so forth. You know, it's a very big problem that people traditionally and culturally wants to take their bodies, dead bodies, or give them parental rights, and so on and so forth. So how do you how can you, what role can you play in educating people to know that there is boundaries, there is risk in trying to make sure they give the parental rights to somebody who died of highly pathogenic diseases?
Okay. If I understand you say how I communicate about the risk with the family to accept the measure or
so I open the floor for a question. Second. Question? Comment? Okay, so in the absence of questions, Dr. Freeman, thank you very much.
Thank you.
So I don't know, is the Dr. Maka around? The line is he? Okay, so we could not communicate with Dr. Maka Chibo, who should be presenting online. So, and in the absence of his presentation, this brings us to the end of this session. Thank you for your time. Thank you for your attention. Thank you.
Right, thank you very much. Hello? Hola. Hello? Yeah, okay, it's working now. Right, so thank you very much, Dr. Aliyu, for chairing that session. We have the next chair in the person of Prof. Shehu Indams. Prof. Shehu, could you please take up? Thank you, sir.
Thank you very much for calling me and giving me the opportunity to chair this. I will call the first presenter in this session, Dr. Aliyu. Dr. Aliyu is from the Center for Mycotoxin and Food Safety, Federal University of Technology, Minna. He will be presenting on the title, Seroprevalence and Assessment of Knowledge, Attitude, and Practices of Camel Handlers regarding brucellosis and consumption of camel milk and urine in East Senatorial District of Yobe State, Nigeria. You're welcome.
Good afternoon. Distinguished ladies and gentlemen, I'm Haruna Aliyu from Africa Center of Excellence for Mycotoxin and Food Safety, Federal University of Technology, Minna, Nigeria, to present on seroprevalence and assessment of knowledge, attitude, and practice of camel handlers regarding brucellosis and consumption of camel milk and urine in East Central District of Yobe State, Nigeria. Oh, thank you. Way of introduction, brucellosis, it has also several names depending on the region. It could also, it's called Mediterranean fever, undulating fever, Malta fever, rocky fever, etc. Brucellosis is an important but neglected zoonosis that causes serious economic losses in livestock and pose a serious public health challenge in human. Infection in animal could occur when animal ingest contaminated feedstuff, when their feedstuff is contaminated with a Brucella organism or through contaminated water. While women acquire this infection mainly through injection of contaminated milk or milk product, or when they come in contact with infected animal or aborted fetal or fetal membrane or discharges from infected animal. Women unpasteurized camel milk previously was mainly consumed by pastoralists, but recently it has gained wider market and commercialization because of its therapeutic effect, and also it's used for prophylaxis in major diseases that are prevalent in Africa, like asthma, diabetes, hypertension, and even in the management of cancer patients. Brucellosis has been reported in many developing countries, like in Nigeria, with up to 13.5% prevalence rate. And this makes it second most important zoonotic disease after rabies. According to OIE, over 500,000 cases of human brucellosis are reported annually. And according to OIE, FAO, and WHO, globally, brucellosis is being underreported. And most clinicians are not familiar with some of these with disease. Even when they are confronted with a patient, it rarely comes to their mind. It's one of the last diseases that come to their mind. Mostly they engage in treatment of malaria, typhoid, and others. Brucellosis in camel, like in other species, is an insidious infection. That is, it hardly provoke any, you hardly see any clinical manifestation. Camels are believed to be a reservoir of this disease. They only acquire this infection from other small ruminants like cattle and sheep and goat. Human brucellosis is a multisystemic disease with broad spectrum of clinical manifestation with varying degree of organ and system complication. It affects different organs like musculoskeletal system, gastrointestinal system, and even nervous system. Camels are used for various purposes, for the production of milk, wool, transportation, and meat. Diagnosis of brucellosis poses a serious challenge to clinicians and even diagnostic laboratories at the same time. Because it's one of these diseases with protein manifestation, so it mimics other diseases clinically. Objective of this research was to assess the knowledge, attitude, and practice of camel pastoralists towards brucellosis in Yobe State, and also to determine the seroprevalence of brucellosis in camels. And lastly, was to determine the intrinsic and extrinsic factor of age, sex, and location that influence distribution of this disease in camel. Methodology. A cross-sectional study was conducted and conveniencing sampling technique was adopted to sample camels. And also, a structured pre-tested questionnaire was administered to camel handlers. Methodology that was adopted. Laboratory analysis of serum samples was analyzed using Ross-Benger plate test and serum agglutination test. Data generators were analyzed using SPSS version 20. Descriptive statistics of proportion were used to describe the variables. Also, chi-square and Fisher-Isaac tests were used to test for association. And statistical significance was set at p-value of 0.05. Result. Majority of herdsmen do not have knowledge about camel brucellosis. Some of them know that their brucellosis affect ruminants, but majority of them doesn't have knowledge about brucellosis in camel. Poor practice towards the infection was reported. They keep mixed animals in the same grazing area and even source of water. And majority of them doesn't quarantine their newly purchased animal before adding it to the herd. You can see that about 96% of them throw away carcasses or dead fetus anyhow, and this can serve as source of risk factor in the spread of these diseases. About 100% of them doesn't take any precautionary measure or basic hygienic procedure, that is protective measure, or cleaning of their hand using soap after handling these animals. Table 1 shows the prevalence of Brucella abortus in camel samples using RBT test and serum agglutination test. From this result, more camels were found to be seropositive to Brucella using RBT tests when compared to serum agglutination tests. This is unexpected. From this result, more camels were found to be seropositive to RBT tests when compared to serum agglutination tests. This is an unexpected finding due to the fact that RBT test is a rapid screening test with relatively low specificity but high sensitivity when compared to serum agglutination test. More camels from Gendam local government show more specificity compared to use rate. But there was no statistical difference between prevalence rate between the two local governments at p-value. Okay. But there is no statistical significance between the prevalence rate in the both local government for both tests. The difference in the prevalence rate between the local government was not statistically significant for both RBT test and serum agglutination test as p-value of 0.05. Table 2 shows seroprevalence of B abortus infection in camel based on sex using RBT test. More females were found to be seropositive for both. More females were found to be seropositive for both tests. This could be due to the fact that females undergo some physiological changes, and also in the female milk there is a presence of erythrode, which make them more susceptible to brucellosis when compared to females, when compared to male. From the result, females show high prevalence rate than male, which was statistical significance. Table 3 shows seroprevalence of B abortus in camel based on age using RBT test and serum agglutination stage. From the result, adult camels were found to be more seropositive for both RBT test and serum agglutination test. This could be due to the fact that more adults has been in the environment, and considering the management system where extensive system is being practiced. And according to Kaltungo et al. In 2018, he said management system is one of the factors that aid in the spread of brucellosis among herds. Because here, the farmers keep both small ruminants and then the cattle. And camel, as we know, they are reservoir, so they acquire this infection from other animals through spillover. Table 4 shows risk factor associated with B camel brucellosis. Age and sex were identified as proportional risk factor for potential risk factor for camel brucellosis and were estimated using odd ratio. Results show that females had 0.57 more chances of getting infected than male. This association was found to be statistically significant. Also, age, the results showed that the adult camel were at higher risk of infection with odd ratio value of 0.07. A statistical significant association was observed for this finding. Conclusion. Majority of herdsmen do not have knowledge about camel brucellosis in the study area. Poor practice towards the infection were reported. Against the prevalence of Brucella abortus infection in the surveyed camels were 42.5% using RBT test and 22.4% with serum agglutination test. So prevalence rate was higher in Gendem local government and lowest in Unisari local government. Female camel had significant high seroprevalence, and adult camel had significant high seroprevalence. Recommendation: the government should institute a national policy, routine surveillance and control program for brucellosis in livestock, including camel, to check the spread of disease in the country. Further studies should be considered to ascertain the presence of B abortus antibody antigen in the milk of camel to ensure wholesome milk for the populace. Public health campaigns should be instituted to increase awareness of brucellosis and an appropriate preventive measure against the disease. I wish to acknowledge the SA partner, WANIDA Symposium team, African Center of Excellence for Mycotoxin and Food Safety, Federal University of Technology. I wish to acknowledge my center leader, Professor Hussein Makun, and his team, and my supervisory teams. Thank you for listening.
Thank you for that timely presentation. The floor is now open for questions. Yes, please. Behind. Okay.
Thank you, Ali, for that presentation. The one thing that got me really interested in the work is where you talk about the urine and its uses, that the urine of the camel. Yes. I don't have any information on that, and I guess it's something I would wish you to shed more light on. As to, you talked about it, is it therapeutic use? And I guess that's why you decided to also look at it in terms of the seroprevalence of the disease. So please, can you shed more light on that?
Yeah.
Thank you.
Thank you for that information, for that question. Even over 500 years ago, in Asia and Africa, camel urine has been used for different therapeutic purposes and then management of certain diseases. Before now, we are thinking probably because we are from Africa, where healthcare system, modern healthcare system, is not reachable for majority of the population, and where traditional medicine is in increase. The users of patronage of traditional medicine is in increase. But you will be surprised that even Saudi Arabia Peninsula, a lot of the patients, especially those that are suffering from female breast cancer, patronize a lot of this camel urine. Even for prophylaxis, even if you are healthy, they use it. And scientifically, there has been a lot of work to find out what is the content, what is there in the urine. And they realize it's contain cytotoxic. There is a substance in the urine that have cytotoxic effect, which can be utilized even from to prevent metastasis of cancer cells. It impaired development of cancer cells. In the north, it's a popular. I have seen families that use this urine for different purposes. Because once you are sick, you want to get solution anyhow. The issue of WHO was warning when the Middle East respiratory outbreak happened in Saudi Arabia, that it's possible, if people are not careful, they could contract this infection through the drink of this camel urine. It's on sale. There are regions, when you go, places like Katsina, it's sell like they sell petroleum in the black market. They will say this one is pure, this one is diluted, and that's how they fix their price.
Okay, I'm also thinking, if that's the case, is it not possible that the urine, as it were, may also be inhibiting the brucellosis?
No.
That is the brucella.
Yes.
As in, will now be also toxic to the organism.
I'm coming. Brucella militensis from small ruminants is the most pathogenic of the all species of Brucella, and it has been isolated from camel urine. It impairs other Brucella organism, but it doesn't really have effect on Brucella militensis, which is most pathogenic of all the species. Thank you.
Okay. Do we have more questions? Yes, please.
Thank you very much. I thank you for the presentation. Is it human brucellosis endemic in your country? And did you do diagnosis for Brucella abortus in human infection cases?
Yeah, Sani, 2019 report from his study, he report high prevalence of Brucella and seroprevalence in sample in his study in Niger State. Even though his study targeted on high-risk individuals, butchers, veterinary practitioners, and then pastoralists, he came out with high prevalence in human, which shows that this disease is going on in the population. And unfortunately, even the medical practitioners are not too familiar with the clinical manifestation of this disease. So anytime patients are confronted with them, is one of the last things that come to their mind. It rarely comes to their mind to be put in part of their diagnostic procedure. Again, brucellosis has been found to cause death in human because acute chronic cases that are not managed can result into endocarditis, which eventually can lead to death. And also, it has been reported to cause arthritis. and several organs and system complication.
Okay, thank you for that response. I'll call for one more question, then we end this session. Do we have more questions? Okay, thank you for a nice presentation. Can we clap for him? The next presenter is Lenguiya, who is a PhD student in the University of Guiba in the Republic of Congo. The topic is SARS-CoV-2 infection in pests from COVID-19 positive households in Brazzaville, Republic of Congo. It's an online presentation. Are you available? Okay. Please present your screen and then present. Thank you. Hello, Hussaina? Hello, we can't hear you. Sorry?
We can see your screen.
Hello, Susanna, we can see your screen. Can you go ahead with the presentation, please? We can't hear you. We can't hear you.
to you later after the next presentation. Is Ibrahim, please, can you come?
Thank you very much. Good afternoon, everybody. I'm told prebiotics a valuable means for the prevention or proper management of nutrition is, as you all know, one of the largest single worldwide malnutrition, as you all know, is one of the worldwide largest single underlying cause of death, child death, especially in low and middle income countries, as the case is in South Asia and most of the sub-Saharan African countries. So malnutrition kind of promotes the incidence, chronicity, severity, and case fatalities of commonly encountered infectious diseases by compromising the immune system and making the infant more susceptible to these infectious diseases. There's a kind of synergistically and bidirectionally linked relationship between malnutrition and infectious diseases that make them a very huge public health problem, especially in countries like mine. So, usually the most common form of this malnutrition, that's the moderate form, is not a life-threatening condition, but is a threefold risk factor to the life-threatening condition, that's the severe form of the malnutrition, and the one that is mostly associated with infectious diseases. So what have been also been noticed with this kind of malnutrition is both kinds are associated with this biosis, because usually they occur in at a period where the child and the infant are seeing the establishment and maturation of their microbiota. And this malnutrition would create a condition that would kind of perturb the proper establishment of the microbiota, meaning that when you treat infants with the commonly used treatment, that's therapeutic flowers, they help, these standard therapeutic flowers help the refeeding process, but the infant remains stunted and at risk of undergoing subsequent episodes of malnutrition because of the dysbiosis prevails. So what currently is feeding scientific literature is that when you want to address malnutrition, that's the best way to go to is to also, as you are trying the refeeding process, also target the microbiota for its amelioration and its maturity. So that's what we are trying to do. it's obvious that the nutritional status of the infant has a crucial role in the prevalence, incidence, prevalence, and outcome of this malnutrition. Surely the associated infectious diseases. So we are trying in a One Health mindset, you are trying to address this malnutrition by producing local made with local ingredients therapeutic flowers that are rich in prebiotics, especially prebiotics that are of polysaccharide source. So the way we go through it is through this project. We still have a long way to go, but at least we've started with this project co-funded by the AS and CE partners. So the way we are going through it is first we've looked up into the dishes of mothers, caregivers, and advisors for the prevention of this malnutrition in three regions of Niger. A region that has a high prevalence of malnutrition, a region that has a low prevalence, and a region that has intermediate prevalence of malnutrition. So what we've been able to gather is what we are doing in the next step, look to assess for the biochemical content and also the prebiotic potentials of these prebiotics that we shall use in the work packet 3 in a formulation of therapeutic flowers that we shall use then in a clinical trial while monitoring the clinical status, nutritional status alongside with the eventual impact these therapeutic flowers would have on the microbiota. So this is a very recurrent and quasi-permanent health issue that we have in our context and that we are trying to address with a One Health approach through addressing the malnutrition aspect because of when you primarily address malnutrition is like you are addressing, you are lowering a risk factor to the infectious diseases that are associated with the complicated, that's the severe form of malnutrition. So this is just an introduction of a new research area that we've started in our lab that could come and complement what has already been explained by my scientific director this morning. Thank you very much.
Yeah, thank you, Ibrahim, for the presentation. Are there comments or contributions to what he has presented? I'm sure it's an interesting area and it calls for interactions. So now that here you can, okay, there's somebody here. Thank you.
Yeah, thank you so much for the presentation. In terms of malnutrition, I don't know if there's any project that has been done looking at the impact of intestinal inflammation and the gut microbiome in the area where you are. Second question is concerning the medicinal plants. Generally, when we were growing up, there are certain plants that are given to children when they are not eating well. I don't know if there has been any investigation, to see if malnutrition can be improved with the application of such plants.
Okay, to your first question, I would say that there are huge literature reports concerned with the inflammation and microbial translocation that is favored during a dysbiosis that causes this malnutrition. And with respect to the traditional plants that you are talking about, is because of that in our first work package, we've been interested in a sociological, socio-anthropological survey to look into the dishes of the indigenous, a kind of ask for the traditional way they used to handle or to prevent this malnutrition. Is a way for us to say, since those people are not having such problem and you are in the same area, so how is it that they are not having the problem? Is it the way they are feeding their children with the same thing that you could have available to you? So if that is the case, so let us have a scientific basis of the usually done traditional process for this prevention and management of malnutrition, and come back to the mothers, caregivers, grandmothers, or advisors, to come back to them with a standard formulation of these flowers that could be homemade, so that everybody in every area in Niger and at any time could have these ingredients and have a homemade of these flowers that could help in the prevention and management of the malnutrition, and by the way help reduce the incidence of infectious disease that could be eventually associated with the severe forms.
Do we have more comments or questions? Okay, can we clap for Ibrahim, please?
Thank you very much.
Are we ready with Hosanna now? It's like you've made yourself up.
Bonjour tout le monde, est-ce que vous arrivez à m'écouter?
Yes, we can hear you.
Ok. Bonjour tout le monde, je suis Osana Lengia, doctorante à la faculté des sciences et techniques de l'université Marie-Ngouabi. Je vais vous présenter la synthèse de mon travail de thèse qui porte sur l'infection à SARS-CoV-2 chez les animaux de compagnie vivant avec des propriétaires testés positifs à la COVID-19 à Brazzaville en République du Congo. Ma présentation comprendra 4 parties. En effet, depuis le début de la pandémie de COVID-19, plusieurs cas des animaux testés positifs au SARS-CoV-2 sont rapportés en Asie, en Europe et en Amérique. Au niveau de l'Afrique, une seule donnée rapportant l'infection d'un puma et 2 lions a été rapportée en Afrique du Sud. Cependant, la grande majorité des animaux infectés par le SARS-CoV-2 sont des animaux domestiques. Au niveau de la région africaine, selon notre connaissance, il n'existe aucune donnée rapportant les cas d'infection des animaux de compagnie par le SARS-CoV-2. En effet, nous sommes sans ignorer que au niveau de l'Afrique en général et au Congo en particulier, il existe un grand nombre des animaux de compagnie et les différentes interactions qui existent entre l'homme et les animaux ne nous permettent pas d'exclure le risque d'infection de ces animaux par le SARS-CoV-2. C'est alors dans ce contexte que nous avons émis comme hypothèse que le SARS-CoV-2 circulant dans la population humaine pourrait être transféré aux animaux de compagnie et ces derniers pourraient servir de relais entre la population humaine et les animaux sauvages environnants. Pour vérifier ces hypothèses, nos objectifs ont été d'investiguer les cas du SARS-CoV-2 chez les animaux de compagnie, de caractériser les différents variants infectant les animaux de compagnie, ainsi que de déterminer la séroprévalence de l'infection à SARS-CoV-2 chez les animaux de compagnie à Brazzaville. Pour atteindre ces objectifs, nous avons mené de février en décembre 2021 cette étude en se servant de la base de données nationale du Congo concernant la COVID-19. Nous avons pu recenser les patients testés positifs à la COVID que nous avons contactés pour avoir leur consentement. Après le consentement des propriétaires, nous avons pu inclure un certain nombre d'animaux dans cette étude en réalisant des prélèvements ainsi qu'en collectant des données socio-épidémiologiques et des données cliniques. S'agissant des prélèvements des animaux, nous avons réalisé en premier lieu des prélèvements des écouvillons nasopharyngés et rectaux en partant du principe que, une fois le propriétaire des animaux testé positif au COVID-19, dans un intervalle de moins de 15 jours, nous l'avons contacté pour prélever son animal de compagnie. L'animal testé positif au premier prélèvement a été reprélevé chaque 2 jours jusqu'à ce que son résultat s'est avéré négatif. Les animaux testés négatifs au premier prélèvement ont été reprélevés 2 jours après pour confirmer le résultat. Vers la fin de cette étude, nous avons réalisé des prélèvements sanguins chez les animaux testés positifs et négatifs aux écouvillons nasaux et rectaux. Comme technique de laboratoire, nous avons utilisé la PCR en temps réel en ciblant 3 régions du SARS-CoV-2, à savoir une région des polyprotéines ORF1AB, la protéine S ainsi que la protéine N. Les échantillons testés positifs ont été analysés en utilisant le système MySeq et les échantillons sérologiques ont été analysés en utilisant la plateforme Luminex afin de rechercher 3 types d'anticorps dirigés contre le SARS-CoV-2, à savoir les anticorps anti-N, anti-S et les anticorps anti-RBD. Comme résultat, sur les 10 mois qu'a duré cette étude, environ 6000 personnes ont été testées positives au SARS-CoV-2 au niveau du Congo, parmi lesquelles nous avons pu contacter 5000 personnes. 185 ont confirmé avoir au moins un animal de compagnie chez eux et 65 nous ont donné l'autorisation pour pouvoir prélever leurs animaux de compagnie. Dans l'ensemble, 100 animaux ont été prélevés pour les analyses de biologie moléculaire et 65 ont été prélevés pour les analyses sérologiques. S'agissant des prévalences de détection du SARS-CoV-2 chez les animaux de compagnie, sur 100 animaux prélevés, 14 ont été testés positifs en PCR et 22 en test antigénique. Donc, dans l'ensemble, les prévalences décrites chez les animaux de compagnie au niveau mondial sont assez variables. On note les prévalences allant de 5,3 à 33,3 concernant la détection de l'ARN et les prévalences de 7,8 à 44,7 concernant la séroprévalence. S'agissant du suivi de l'infection des animaux de compagnie, 37% des animaux ont présenté des signes cliniques liés au SARS-CoV-2 comme on observé chez les humains, à savoir les éternuements ainsi que les difficultés respiratoires. D'autres signes cliniques, comme les signes cliniques relatifs aux gastro-entérites, ont été également notés chez les animaux. Les animaux ont excrété le virus pendant une durée allant de 1 à 12 jours pour les chiens et de 1 à 17 jours pour les chats. Quant aux différents variants détectés chez le SARS-CoV-2 chez les animaux, sur 14 animaux testés positifs par PCR, 10 ont pu être séquencés et les séquences obtenues correspondaient aux variants bêta, aux variants localement appelés congolais, ainsi qu'un variant recombinant entre les 2 variants précédemment cités. Les variants décrits chez les animaux correspondaient aux variants ayant circulé pendant la période de novembre 2021 en février 2022 dans la population congolaise ainsi que dans la population des pays voisins. Pour conclure cette étude, nous pouvons dire que l'infection à SARS-CoV-2 peut bel et bien avoir lieu en Afrique ainsi qu'au Congo. Et les différents variants détectés dans cette étude ont aussi été détectés chez la population humaine. On peut dire que les animaux peuvent excréter le virus pendant une durée de plus d'une semaine. Sur ce, me voici à la fin de cette présentation. Je remercie toute l'équipe d'organisation de WANIDA pour l'opportunité qui m'a été accordée de présenter ces données et je vous remercie pour votre attention.
Thank you very much for that presentation. The floor is now open for comments, questions. Are there questions, please? Okay.
Thank you for the presentation. I'm interested in knowing if the type of COVID variant that is found in the animals, if it's the type that can be transferred to human. I'm not sure I got that during the presentation. Thank you.
Merci beaucoup pour votre question. Pour essayer de répondre à cette thématique, à ce jour, il existe seulement 2 cas de transmission inverse des animaux de compagnie à l'homme qui ont été rapportés. Mais notre étude ne concernait que la partie de transmission de l'homme aux animaux de compagnie. Nous n'avons pas pu analyser le volet de transmission inverse des animaux de compagnie à l'homme.
Thank you.
Do we have more questions? Yes, please.
Merci pour votre très brillante et intéressante présentation. Le sujet m'intéresse très bien. En fait, la question, peut-être je vais poser autrement la question de notre collègue, est celle de savoir si les variants ou les génotypes, c'est pas, que vous avez trouvés chez les humains, est-ce que c'est les mêmes que vous avez eu chez les animaux infectés? C'est la première question. Des deux, comment pouvez-vous prouver que ce n'est pas plutôt la transmission inverse qui a eu lieu? Est-ce que au moment où vous avez prélevé l'animal, comment savez-vous que ce n'est pas lui qui était infecté en premier?
Merci beaucoup pour vos questions, messieurs. Pour essayer de répondre à la première question, je peux dire que comme je l'ai dit j'ai essayé de le dire dans ma présentation, les variants détectés chez les animaux de compagnie ont aussi été détectés chez les humains dans la même période de notre étude. Et s'agissant de la deuxième question, concernant entre l'homme et l'animal qui a été le premier à être infecté, pour la plupart des enquêtes que nous avons eu à effectuer, les humains ont rapporté avoir commencé à développer des signes cliniques de l'infection bien avant leurs animaux de compagnie. Donc sur cette base-là, sans aller plus loin, nous pouvons dire que l'infection s'est faite plutôt des humains aux animaux de compagnie. Mais étant donné que nous n'avons pas eu à analyser dans le cadre de cette étude les échantillons des humains, nous ne pouvons pas affirmer de façon très objective que les animaux infectés ont bel et bien été infectés par leurs propriétaires ou par des voisins. Merci.
Okay, thank you for that response, Hosanna. Do we still have more questions for her? If there's none, then we clap for Hosanna, please. And with this, I've come to the end of my chairmanship, and I hand you back to the organizers. Thank you.
Right, thank you very much, Prof, for chairing this section for us. So I think we are all tired, we need to stretch, and it's time to eat. So we'll go for lunch, and we'll come back for our next session. So we have 1 hour break, and we all head towards the restaurant, and then we can have something to eat. So enjoy your meal. Bon appétit. Right. So yeah, 1 hour we come back. Yeah. As usual, please leave your headsets behind. Don't take them with you. Kindly leave them on your tables or chairs, and then when you come back, you pick them up. We don't want to lose any, so please help us keep them all safe. Thank you.
So
2 o'clock. Time is over.
So good evening, everybody. We are a bit late, but we have to wait because people are still eating. So we will give them little minutes to come. I hope you enjoyed the lunch.
Bon, attention s'il vous plaît. Please give me your attention. Right, so welcome back from lunch break. And now we're going to the last part of our program for this symposium. So we're going to have the theme Insights into Capacity Building and Resources Sharing. And we have the honors to have our own Professor Mbaye, who is going to share this section. So please, let's welcome our chair. Thank you.
Bonjour à tous. Je m'appelle professeur Maïssa Mbaye. Je suis le coordonnateur du Centre d'excellence africain en mathématiques, informatique et TIC. Et je représente aussi le réseau des centres d'excellence en sciences et technologies du numérique. Je parlerai en français pour aider aussi les traducteurs à travailler. Alors, dans cette session, nous allons avoir deux parties. La première partie est constituée de deux présentations que nous allons avoir, de deux structures. Déjà, on va avoir une présentation venant du Crick Institute. Ensuite, nous allons avoir une présentation venant de Gabriel International Research Center. Donc là, ce sont des présentations qui vont nous permettre d'introduire le sujet autour des réseaux numériques ainsi que leurs bénéfices en termes de renforcement de capacité. Ensuite, nous aurons le panel ou la table ronde numéro 3 qui vont nous permettre d'en savoir un peu plus sur les leçons apprises et nous aurons l'occasion de revenir avec tous les intervenants. Donc, sans tarder, comme nous sommes un peu en retard, je vais introduire Dr Bauer, donc David Bauer, qui va nous faire une présentation sur les plateformes temps réel de virologie et immunity, les leçons qu'on a apprises du Covid-19 de Londres, de l'ouest de Londres et au-delà. Donc, M. Bauer est un virologue et est le responsable du RNA Virus Replication Laboratory au niveau du Francis Crick Institute à Londres. Donc, M. David, vous avez la parole.
Merci beaucoup, Prof. Mbaye. I will present in English, mais en français. Tout d'abord, je tiens à remercier les organisateurs, le réseau WANIDA, l'équipe ACE Partner et nos autres organes au WACPIP. Merci pour l'invitation et pour l'opportunité de présenter ici aujourd'hui. I will present in English. Thank you again to the organizers for the opportunity to present and speak. I'm going to talk briefly about our experiences at the Francis Crick Institute in London. And of course, while these might seem to be a world away from other countries in the world, and in fact they are often a world away from other laboratories within the United Kingdom, there are some really important lessons that we've learned about the possibilities for us as researchers to contribute immediately and directly in the course of a pandemic. I will talk at the end about some new collaborations that we founded and some of our hopes for capacity building in terms of how to approach pandemics in real time. So, as a brief introduction, for those of you who were not able to see Professor Langhorn's talk this morning, the Francis Crick Institute is a large research institute, allegedly the largest in Europe. And you can imagine it's like the entire US National Institutes of Health compressed inside one very large building, maybe the size of a football stadium. And it goes down underground as much as it goes up. And we're in the center of London, right next to the Eurostar terminal. So we have lots of opportunities to get around. and escape the little island that we live on. It's a beautiful facility, it's huge, and there are people working on everything from cancer to neuro things to biophysics, and of course immunity and infectious disease. We host one of the 6 WHO collaborating centers for influenza, which is kept busy updating the flu vaccine. In fact, they receive lots of samples here from Ghana. And we also have a set of science and technology platforms, which are core facilities where they function not just to provide services, but they also have experts in these techniques, almost like a technical laboratory, to support research not only for the technique, but to collaborate with everybody else. And you've heard a bit about the CRIC African Network and how that works this morning. So when COVID-19 came to the CRIC, I didn't work there. I only joined about 6 months into the pandemic. But my lab looks very similar to the rest of the Institute. So we are about 9 people, and these people come from very different backgrounds. So yes, there are virologists, but there are also people who are molecular biologists who study RNA. They're chemists even. We have people who have immunology backgrounds in working with mice and with larger mice called people. And we've spent a lot of time working not just on our own about COVID-19, but collaborating with a study that the CRIC set up called the Legacy Study. And this on the ground is led by Dr. Emma Wall, who is a consultant in infectious disease, by Mary Wu, who is an expert in automation and robotics. She is not the robot, but and Ed Carr, who is a nephrologist, a kidney doctor, who has a strong interest in immunology and vaccines. And so together, along with all of our colleagues at the Francis Crick Institute, we've been spending lots of time to understand COVID and immunity. The first thing that the CRIC did was to set up PCR testing in March 2020. And the CRIC did this as a large effort together with its partner hospital at University College London. And at that time, in March 2020, there was no testing capacity available in the UK whatsoever. But like all big cities in the world, or at least within Europe and the US, they got a very large wave of COVID very quickly. And so the CRIC partnered together with the hospital and with a diagnostic laboratory to set up PCR testing very, very quickly. And so the government was not doing much of anything at that time. They were thinking about setting up very large facilities, and the CRIC simply did it. At the end, we ended up conducting nearly 750,000 tests, serving 9 public hospitals, 89 care homes for the elderly and disabled, which of course, in that environment, people are in very close contact. It's very hard to avoid COVID, so it takes a really good effort to test people and quarantine them. 1 mental health trust, which has similar situations, as well as 4 research institutes to allow people to come back to work. What was most interesting to me as a scientist is that if you look at this graph of all the tests that were done, so most of them, of course, are negative, and that's what's shown in green. But even just us, by the time the government started doing testing, we were a very small piece of the puzzle. But down here, you can still see the waves of the pandemic. So this was what became known as the Alpha variant that hit us. At the same time, Southern Africa was experiencing what became the Beta variant, and South America was experiencing what became known as the Gamma variant. We were able to take these samples for the purposes of surveillance and sequence them together with a large British consortium called COG UK. And what's most striking is that even in our tiny little corner, in our little research institute, little compared to a large national laboratory, we were able to follow the entire path of the pandemic. So we were like a little microcosm, a micro version of society in London. And so you could see each of the variants as they came along. And so as a result of that, the CRIC decided together with its hospital partner to set up a research study for the next phase of the pandemic. So that is, okay, we are all going to get vaccinated, or if not vaccinated, infected, and hopefully recovered. What happened with people's immunity. And so the question for the public health point is, do vaccines still protect us? So we have now 821 adults in our study, and people were subjected to weekly PCR testing, not optionally, but mandatory as part of the institute and the hospital's policies. So we know when people had COVID, and we know what variant they had. We know what vaccine they had and when they had it. And since they volunteered for the study, we also are drawing blood regularly, so we can look at PBMCs and at serum. And so this provides an extremely rich set of data to understand what's happened to these different people. Is there a difference if the first variant you encountered was Omicron or Delta? Did it matter if you were infected before you were vaccinated or after? Now, as you can probably start to imagine the multiplication in your head, 821 people, maybe 8 or 10 samples just of blood alone, that starts to become an unreasonable amount of work and very expensive for reagents. And so for this, we turn to the science technology platforms at the CRIC, who were able to miniaturize a live virus neutralization assay within high containment labs. And so what ends up happening is in 384 well format, we are able to test whether serum is able to neutralize one variant versus another. And this is a much more sensitive method for detecting or predicting protection from infection than an ELISA, because an ELISA is telling you about sticking, but not if it neutralizes. At the same time, we are able to look at infection dynamics, so how do people's viral loads go up and down? And as a result of this, we've ended up publishing a series of papers looking first at people who'd received the Pfizer vaccine as the Delta wave started to take off. But critically, there were no studies at that time about people who had received the Oxford-AstraZeneca vaccine, which of course is the basis for the COVAX program that's been used. And so we put out a call within the CRIC to ask people who had received the Oxford vaccine to please volunteer. And within 2 weeks, we had bled over 150 people and put them through this pipeline. And we were able to go back and confidently say that people were still going to be protected, especially if they had been previously infected with SARS-CoV-2. This, of course, has continued for the new variants, and as well, we are trying to come up with an understanding of how monoclonal antibodies protect people. So the outcome of this for us has not just been nice publications, but real-time data sharing. We put everything online. We put all the raw data that we can onto GitHub. People have taken this data to do modeling. We, of course, pass it to the government. These are acronyms for important people in the UK, like the Chief Medical Officer. We've published lots of papers and also had an impact on policy. But most importantly for us, of course, is now the pandemic, right? If for practical purposes, it's over. If you are extremely vulnerable or sick or immunocompromised, it's still a problem. But for our daily lives, I mean, we are here, we are traveling. What we want to do is partner with other people within the CRIC and without, with a focus on trying to do discovery science, because that's what we do. We are fundamentally about understanding why. Why do things work the way they do? Of course, applying that to policy is really important for public health, and also translation. So can we improve medical practice or policy practice in an applied way? And so one of the key questions that we all have heard about yesterday, both from Yao Bediako in his talk about Yamachi and biotech, which he mentioned at the end, and Peter Kwashi's work at WACBIP, has been trying to pick apart why was COVID so different in West Africa. Of course, there were lots of lockdowns, and there were lots of severe consequences for society. But actually, the experience was very different from what we experienced in London and what others experienced in India, China, North and South America. And so we've established a consortium, first bringing together the legacy study at the CRIC and the heritage study led by Yamachi Biotech and Yao. And Yao's study is completely independent. It's his idea, it's his baby. But the important question still remains. What were the differences? And so by comparing things together on the same platform with standardized methods, we hope to gain some insight. But of course, that's not going to convince the rest of the world, because people will simply shrug and they will say, ah, yes, but you know, West Africans are different somehow from people in London, and you don't really know. Is it environment? Is it genetics? Is it some combination? Populations are different. People are younger. People are older. And so what we looked for was perhaps something that could serve to as close as a control as we can find. And that, of course, lies in the West Indies, in the Caribbean. And so there is another study, which they have renamed Windfall to line up with our naming, which is very similar, studying adults in Jamaica who are healthy before they were vaccinated, after they were vaccinated, with serial drawing. And of course, in Jamaica, we have overlapping genetic ancestry between all three locations. We have very similar vaccination regimes to West Africa and West London, and roughly similar course of the pandemic, one variant than the other. But in Jamaica, the experience of COVID has not been like West Africa. In fact, it's been very similar to that of the US or West London. So why is it that in that situation, where the age distribution is roughly similar to West Africa or India, why did the outcome look more like India or the US than it did West Africa? And so we've put together a consortium bringing together the University of the West Indies together with these studies, as well as WACBIP here. And in London, the London School of Hygiene and Tropical Medicine for doing computational modeling, and our clinical hospital partner who's run our study. And so we have funding now from the Wellcome Trust to start this and get going. So we are just starting. And we have key questions. One is, of course, what I told you. Why was COVID so mild? And there are 4 hypotheses. One is maybe it was a little bit different. So I said it was all the same. Maybe I'm lying a little bit. Maybe there were slightly different variants which set you up for a different immune response when a nastier one came later. So we will check. We will check serum from all different locations with the different variants and see if there is some better ability early on to neutralize one versus the other. The second might be that people have different exposures to circulating natural coronaviruses. So bats, for example. I mean, if you step outside here in the evening, there are lots of bats flying around. London, not so much. In the West Indies, not quite as much, depending on where you live. The third one, of course, is almost too obvious. In fact, Professor Awandare has told me very certainly that it will end up being number 3. But he is, of course, a malaria biologist himself, so I would imagine that would be what he would say. So the one big difference, of course, in West Africa is there is endemic malaria. There is not in Jamaica. In fact, malaria was eradicated in the 1960s. There is, however, sometimes importation of malaria from Haiti to Jamaica. So there will be some people in this cohort who will be positive. It will be interesting to see what they find. And the last one, of course, is somehow a different immunoreactivity. So does pathogen burden overall contribute? Does the level of inflammation response somehow contribute? And again, we have heard in the talks in the past 2 days some hints that that very well might be the case. So we will be able to genotype people, to carefully control things, to try to address these things. And the second thing we will do is harmonize our studies and transfer capacity, because this is not about the British Museum model of science, where you send everything on a plane to London and you never see it for the next 300 years. The first is we are transferring our live virus assay to WACBIP, because there are, of course, CL3 facilities, BSL3 facilities at the Noguchi. We actually are not set up to do pseudovirus neutralizations. That's something that Peter Kwashi has spent a lot of time getting set up, and he is going to transfer that both back to us so that we can learn how to do this assay, as well as to the University of the West Indies. In turn, the University Hospital of the West Indies does lots of diagnostic testing, so on sort of a level where the information is not quite as rich. But if we build up large cohorts, we want to see, can you actually squeeze information out from this data? And so they are going to pass that back to us, and to the team at the London School of Hygiene and Tropical Medicine, who is transferring modeling to the University of the West Indies in Trinidad and Tobago. And so as a result of this, we will have standardized data, same methods, coordinated with same assays, which I won't talk about too much. But basically, we do these in 96 well format for neutralization. This is in CL3. You can, things went quickly, sorry. We can scan them on a microscope. We have an automated web-based system for interacting with all of the data. This can be boxed up in a little container. You can run it on a laptop. You don't need a crazy system to do this. And all of this is calibrated to the WHO international standards. So instead of simply reporting on a set of data from location X, where people will shrug and say, oh, I don't know what that y-axis, what does that number mean? You can actually calibrate your results. We get very, very high throughput, about 2,500 samples per week. And critically, we've set up a set of open source standards. So we are going to release this to anybody who wants them. You don't need to access the WHO's samples for the international standard. We struggle to get a hold of them. God help anybody else. They are like 5 kilometers up the road from us, and we still can't get them. So we've created synthetic standards that if you can express proteins in E. coli, you can make these yourself. They're stable, you don't need to do anything very fancy, and it allows you, as you can see in the middle, to run an assay 1 year later and still get the same results. Critically, you don't need even a microscope. On the right-hand side are curves generated with a plate reader, so you don't need some fancy imaging platform either. So I will just end by saying the ultimate aim is to take these models that we develop, share them out, to the individual sites first, who will say, can we predict immunity and immune responses? And how much data do we really need? Because of course we don't clearly need to do all of it, but it would be nice to know how much information do you need, not in London, but in different settings. I will end now. And just thank the partners, Professor Awandare, Yabediako, Peter Kwashi, in the West Indies, Christine Carrington, Josh Anzinger, and in West London, Emma Wall and Adam Kucharski. And of course, all of our partners, and thank you for your attention.
Merci beaucoup pour cette brillante présentation sur cette recherche très intéressante. Moi, j'ai des questions, mais je vais d'abord laisser la parole au public pour des questions. Nous avons le temps pour 3 ou 4 questions. Une question? Oui, là. Oui, ils vont vous apporter le...
Thank you very much for that beautiful presentation. Because all the work that we do must translate to patient management. So I'm so much interested in the study where you show 821 participants. And then, if I get you correctly, you enrolled them pre-infection, is that correct?
They've been enrolled either pre-infection, but after vaccination, most of them, because they were being vaccinated very early in the UK.
Okay.
Or after infection, because they were allowed, if they had a positive test, to also join.
Okay. The first thing is this. Out of those 821 that you follow for almost 2 years, do they all complete that 2 years? If they do not, did you do risk assessment on them? And then if you do, were you able to identify any factors that are associated to the outcome that you classify as risk? Thank you.
Thank you for the question. And one of the problems of doing observational research is it's not like test tubes or lab mice. The people do what they want to do. Some of them have left the study. Some of them have left the crick. They moved on to other jobs, so we can't follow them up. None have died, thankfully. These are mostly healthy working age adults, and so we are not recruiting in the hospital setting from patients who are very sick. We are just recruiting from the staff. We don't know clearly enough what the risk factors are, but we can see that there are some innate features about people in how they respond to vaccines. So you might imagine that you have immunocompromised people. They would respond badly to vaccination or infection. And you might have people who have been vaccinated and infected, and they would have super immunity. But actually a normal, healthy adult tends to have 2 kinds of responses. They either respond quite narrowly, to the vaccine, or they respond quite broadly and they're protected against lots of different variants. And so we're trying to understand exactly that. This isn't a risk factor for severe illness, but it was a risk factor for whether or not you got BA2 Omicron. So if you are in this broad responding group, you didn't get it, more likely. And so we think this has something to do with how people's immune systems are responding, and hopefully we will find some factor, but we don't know yet. It's a good question.
Merci. Une dernière question, peut-être? Vous avez levé la main? Ok. Une dernière question? Alors moi, j'avais une question par rapport à vos données. Est-ce que vous les mettez en open data pour que des chercheurs de différents secteurs puissent les utiliser? Et l'autre question, c'est une question de curiosité, c'est quoi l'immunoréactivité? C'est en tant qu'informaticien.
So I will answer the first question. All the data, was it a question?
Yeah.
So all the data that we've put online, you can find in the appendix to our papers. So there is a GitHub link. You can go and you can see that people are forking it and doing stuff. And we can see that there are some publications from groups in India and Australia where they've just taken the data. And this is the point. We remove any identifiable information, so you can't look at it and figure out, oh, that's Professor so-and-so in the study. But so we think that this is the way forward. I mean, we are funded by charities and by taxpayers, and there's no reason to hold the data. It doesn't help anybody. So we think this is really important. The second was about immunoreactivity. So one of the things that's kind of clear as I answered to the previous question is that people react differently. Even normal, healthy people in London, where people's infection history is quite homogeneous. And if you extrapolate from that, you wonder if people are living in environments where they encounter different pathogens throughout their lives, not even different numbers, but just different ones, whether this sets the register for how they will respond in the future. And of course, people are never directly comparing with the same methods, with collaboration, within the same network, the same approaches. And so it's easy to ignore and not get an answer and to dismiss for technical reasons when people make comparisons. So we think it's really important to work together for this.
Merci beaucoup. Je vous demande de l'applaudir.
Merci.
S'il vous plaît. Merci beaucoup. Alors, nous allons passer sans tarder à la deuxième présentation de Madame Florence Koumarian Pradet. S'il vous plaît. Donc, elle est responsable head manager de GRABIEL, la Global Approach Biological Research on Infectious Epidemics in Low Income Countries, au niveau de la Fondation Mérieux. Vous avez la parole pour 15 minutes, le temps de nous présenter.
Merci beaucoup. First, I would like to thank the WANIDA organizer for the invitation and for giving me the opportunity to present the Gabriel Network. So, I am Florence Koumarian Pradet from Fondation Mérieux, and I am in charge of the coordination of the International Scientific Network, which is named Gabriel, which serves capacity building in research and surveillance on infectious disease. So, Gabriel Network is a network of 22 research institutions located in 3 different continents, in Asia, Africa, in South America. So, as I said, it's coordinated by Merieux Foundation. So, Merieux Foundation is an independent family foundation which was created in 1967 by Dr. Charles Merieux, and we have a public interest status. We are a foundation, an operating foundation who work in the field side to side with our partners. And the mission of the network is to enhance local applied research capabilities. So by improving laboratory and hospital-based surveillance and research capacity for infectious disease that have a major impact on public health. To strengthen also capacity to conduct clinical research, and also faster assessment of innovative diagnostic tools. So here you have the map of current member. As you can see, we have members so in Caribbean with Centro Gestio in Haiti. We have 3 members in Brazil. In Paraguay, you have also a lot of members in Africa, in Cameroon with the Centre Pasteur of Cameroon, in Madagascar, in Republic of Congo, in Democratic Republic of Congo, in Mali, and also in Lebanon, in Georgia, and in Asia. So our members are either linked to universities, or linked to hospitals, or are linked directly to Ministry of Health. Some of them have are national reference laboratory, for example, for flu in Brazil, on leprosy in Madagascar, or on TB in Georgia. So we have different types of activities within Gabriel. Activity for the coordination and the structuration of the network, activity for the capacity building, and activity to implement collaborative research projects. For our first activities, in order to strengthen the South-South collaboration, we initiated a few years ago a call for proposals. That means that the members work together, at least 3 members of the network, to propose a common project in order to launch pilot study, and after that to be able to search for external funding. Another point that is very important for the network is our international meetings that occur every 18 months. The next one will be co-organized with Centre Pasteur in Cameroon in May of this year. We also started to organize a lot of webinars, especially during the COVID-19 pandemic, when it was not possible to travel. Concerning the capacity building, training workshops, we organize a lot of training and workshops on site. And for example, in last December, we organized a workshop on bioinformatics in Senegal. We also develop e-learning training. We are currently developing with FOUND a training on bioinformatics. We have also a program to support the next generation of researchers by providing a doctoral fellowship. We have also started this kind of program with ERD last year to provide also some scholarship for Gabriel members and to allow a partnership with some Center of IRD and Gabriel members. We also organize during each international meeting of the network a Young Research Award, which allows to give 2 prizes of the best presentation of the young scientist. And also, during the COVID pandemic, we have also provided a lot of capacity building for diagnostic, surveillance, and characterization of the variants. And also, we implement research projects, but I will discuss this later. Concerning the early response that we have provided since the beginning of 2020. In fact, all our members were very mobilized for the diagnostic activities, and we provide a lot of support for them to start their diagnostic activities. At the time, there were no commercial kits available during this time, so we provide, for example, primers that has been published by La Charité. We provide enzymes, positive control, thanks to the European network EVAG, which is a network with a great virus collection. And so this allows to start diagnostic, COVID-19 diagnostic. We also implement some research capacity concerning COVID-19 with a study on nosocomial transmission of COVID-19 in different places, in Mali, in Bangladesh, Madagascar, etc. And we also mobilize some skills to track SARS-CoV-2 variants. And when we start these activities, we organize a mapping of the capacity within the network at the beginning of the pandemic. And here you can see all the steps that are needed, from the sample collection to the sequence data analysis that is reported to GSAID. And what we saw that there were a lot of missing skills at this time, missing skills in bioinformatics, the availability also, the access of reagents and consumables was very difficult. There is also some gap in IT infrastructures, human resources were not sufficient, and also there was a problem in access to sequencing platform. So we decided to provide support for the detection of variant of concern by a stepwise approach with different partners, with 2 partners in Bangladesh, with Lebanon, with a partner in Cameroon and Madagascar. So the first step was the implementation of variant screening by RT-PCR. It's a targeted PCR that detects specific mutation or deletion of the variants. And this was done for routine diagnostic. The advantage is it is very quick to obtain the results. Limited resources are required, and it is low cost. There are also, of course, some limitations. It's sometimes difficult to discriminate between variants, and is not suitable for a detection of new variants. So that's why we also work on the second step of our approach, is to perform whole genome sequencing with different sequencing platforms. And this allows us to build capacities for NGS library preparation. Sequencing was either outsourced or was done internally. And also we build a lot of capacity in terms of bioinformatics for sequence analysis. So now we have different type of activities, research activities within the network, and we focus on topics that are public health interest. And these topics have been selected by according to the needs of the members, but by a bottom-up approach. So we have 4 main activities that federates the network: acute respiratory infections, tuberculosis, antimicrobial resistance, and now also SARS-CoV-2. So we work a lot, our members work a lot with vulnerable population, such as refugees, mother and children, and people living in remote area. So we have different kinds of research. It could be clinical research, operational research, surveillance with One Health approach for antimicrobial resistance, and also surveillance and characterization. And in the context of a research project, we perform a lot of transfer technology. For us, training is very important, and it's also the opportunity, for example, for the TB topic to evaluate new innovative diagnostic tests. We have also a program of quality assurance, which allows to organize EQA, to use harmonized SOP, and to also perform audits when there is some clinical research study that are implemented. This slide shows you just the number, the distribution of research topics within the network. As you can see, 15 members works on COVID. After that, there are a lot of members working on acute respiratory infections, on AMR, and tuberculosis. And now I will give you some example of what kind of study, collaborative study are ongoing in the network. So I will start with the pneumonia. Pneumonia accounts for 15 of all deaths in children under 5 olds. It can be caused by viruses, bacteria, or fungi. It can be prevented by immunization, by adequate nutrition, and it also can be accurately treated if it is well diagnosed. However, more than 40% of pneumonia cases are inappropriately misdiagnosed, especially in LMIC. So we have 2 ongoing projects. The first one is a prospective study on pneumococcal colonization in children and adults that is running in Cambodia, in India, and Paraguay. And the objective of this study is to measure the colonization rate of Streptococcus pneumoniae and what are the serotypes, the predominant serotypes that circulate in children under 5 years of age, and that have already, the countries have already introduced the PCV, which is the vaccine. A second study is done on acute respiratory infection. Wow. in refugee camps to improve and adapt patient care in the case of humanitarian crisis. We work also on TB to improve the diagnostics and management of tuberculosis among the most vulnerable population. As you can see, there are more than 1 million children under 5 with TB disease, and there is a problem for the diagnostic for the children because the collection of sputum is very difficult in this population, and the performance of the tests are not so good. So the aim of the study is to evaluate the performance of 2 blood tests, RISC-6 and a gene based on host response to allow the triage and diagnostic of children on extrapulmonary tuberculosis. And this will improve patient management through non-sputum testing and the proof of concept for molecular diagnosis of TB that can be decentralized also at the community level. Now the last topic, it's antimicrobial resistance. As you know, it's a new pandemic, a silent pandemic. It's one of the top 10 global public health threats. It's a major concern, especially in LMIC with around 700 deaths every year linked to resistant infections. If the trends continue, AMR will result in over 10 million deaths per year by 2070. So we have different activities, and just to mention that we have started to implement the WHO statistical protocol, which is One Health surveillance, which target only one pathogen, resistant pathogen, which is Isbel E. coli, in humans, in the community, in animals, and in environment. And this allows to have data, baseline data on ESBL E. coli prevalence, and allow to have a kind of monitors to assess the impact of future AMR intervention. We have also now some members that integrate international consortium funded by GPIMR, which allow to upscale the tricycle protocol. We have also a fund from Bill and Melinda Gates Foundation that allow to develop a data science center for AMR. This will allow to build capacity for AMR data collection, to process the data, to analyze, to visualize, and to disseminate, especially for the stakeholders, for the Ministry of Health in particular. And we will start a One Health surveillance in the community to target pregnant women and their household, and to assess risk factor for ESBL E. coli carriage in the close environment of the family, which represent a complex One Health network. And now just to thanks to all our members, and thank you for your attention.
Merci beaucoup et désolé de vous avoir mis la pression.
Non, c'est pas grave.
Alors, comme nous sommes rattrapés par le temps et qu'elle intervient aussi dans le panel, donc nous allons réserver les questions pour plus tard. Donc je vous prie de me rejoindre. Donc nous allons passer à la prochaine session qui est une table ronde autour des expériences qui ont été acquises et les leçons aussi apprises sur les réseaux scientifiques. Donc je voudrais bien inviter Dr Mariama Diallo pour nous rejoindre. Ensuite, professeur John Langon et professeur Yap Boom 2. Et ce sera... Peut-être si nous pouvons aussi avoir un deuxième micro. Merci beaucoup, Gaussou. Alors, donc là, c'est un réel plaisir de monitorer de chairer cette session qui est un prolongement de ce qui se fait donc, par rapport aux objectifs de WANIDA. WANIDA a pour principal objectif de permettre la collaboration en termes de recherche pour améliorer la qualité pour tout le monde et pour permettre aux chercheurs de bénéficier des occasions de collaborer avec d'autres chercheurs du même domaine et aussi au niveau pluridisciplinaire. Donc si vous voulez, l'objectif de cette table ronde, c'est de voir quelles sont les différentes approches qu'il y a eu avec différents réseaux qui sont représentés. Je prierai à chaque intervenant de se présenter avant de commencer lorsqu'on va intervenir. Donc nous avons le réseau WANIDA, nous avons le réseau Gabriel, CRIC et le réseau Pasteur qui sont présents pour partager avec nous sur ça. Donc, alors sans tarder, je vais d'abord commencer par Dr Mariama Diallo par rapport à sa question. Donc pour d'abord savoir est-ce que vous pouvez nous décrire brièvement en 2-3 minutes comment est-ce que votre centre bénéficie du fait d'être dans WANIDA?
Merci beaucoup. Merci beaucoup, professeur. Donc, je vais commencer par me présenter. Donc, je m'appelle Mariama Sadio Diallo. Je suis médecin immunologiste. Et je suis maître assistante à la faculté des sciences et techniques de la santé de l'université Gamal Abdel Nasser. En plus, mes domaines de recherche, c'est surtout le VIH, les hépatites, également la méningite et maintenant avec l'avènement du SARS-CoV. Donc on travaille également sur le SARS-CoV. Je suis également la directrice générale adjointe du Centre d'excellence pour la prévention et le contrôle des maladies transmissibles. Donc si on me demande ce que WANIDA m'a apporté, je pense qu'il y a déjà la preuve, c'est que je suis là. Donc je pense que c'est par là en fait, je suis déjà là. Je pense que sans WANIDA, je ne serais pas venue au Ghana, parce que je n'ai pas de relations privilégiées, je n'ai pas de connaissances. Mais le fait d'être dans le réseau, cela m'a permis de venir ici, mais également de nouer des partenariats, des études, des projets multicentriques et d'être associé à des travaux scientifiques, ce qui est très bien pour un chercheur. Donc, je pense que c'est l'un des points les plus parlants. Deuxième point, c'est que je suis venue avec 3 PhD et une étudiante en master pour permettre à ces étudiants-là également de se faire leur propre réseau. C'est-à-dire de discuter avec des étudiants venant d'autres centres et d'établir leur répertoire en vue de l'avenir, parce qu'on construit l'avenir dans le partenariat, dans la collaboration. Et cela, c'est important. Je pense que le partage d'expériences au niveau régional est la clé vraiment de la réussite de l'Afrique. Mais également, et cela va nous amener à des associations avec des chercheurs de l'étranger. Mais je pense qu'entre nous, on doit déjà pouvoir collaborer dans les domaines et mutualiser. Je discutais avec les miniers. La santé et la mine, c'est quelque chose qui va ensemble. Mais je pense que c'est maintenant qu'on va commencer à prendre conscience qu'il y a des thématiques, qu'il y a des choses à faire dans ces domaines. Donc je pense que le réseau ACE, c'est vraiment de mettre des chercheurs africains ensemble, mais également le réseau WANIDA va permettre également les chercheurs des mêmes domaines de mutualiser les acquis.
Merci beaucoup pour cette réponse. Je pense qu'en tant que centre bénéficiaire, ce sont des aspects importants. Alors, je vais passer la parole à professeur John pour dire que Cannes a été vraiment un réseau qui a beaucoup réussi. Je voudrais en quelques mots que vous nous dites qu'est-ce que vous pensez être les facteurs importants de succès et comment pensez-vous pouvoir répliquer ce succès ou bien l'étendre.
Yes. Thank you. What's the reason for our success? Well, I think one thing is very clear from what we've done. We're 6 partners and we all thought exactly the same thing. That is, we identified a gap in where we could help training people, or not really training because we're talking about experienced scientists, but helping people bridge the gap between being a postdoc and an independent investigator in Africa. And I think that's really important that we're appealing to the African diaspora and those in Africa to remain, and we're giving them a limited amount of help, but enough help to let them establish themselves. And I think we all agreed on that point. We would not have been successful without having 18 unbelievably fabulous fellows. And I think they just represent the potential of students and postdocs in Africa. They were absolutely superb, every single one of them. And because they're at this stage of their careers, they're already driven. They're driven to go to independence and don't need guidance. It's all self-motivation. And they motivated themselves and they interacted with their cohort and they motivated each other. And they've all been amazingly successful. I can't tell you how proud we are of them. And I think that's what's made it successful. But by picking postdocs at this stage of the career, you're almost bound for success because they're so driven. And I really do hope that we've helped place people into a larger infrastructure. The other reason why I think we've been successful is our aims actually are small, defined or even fairly limited. We wanted to address one aspect in a way we could measure it with the skills that we have. We can't change the infrastructure of African research. We can't change the global problems we have with sustainability. We can only do what we can do, which is basic science and teaching people to do it properly and help them move their career on. And I think that has been evident. We haven't defined measurable outputs. How did they publish? How did they do their research? Did they get a better job when they finished? Are they still in Africa? So these are limited but defined endpoints. And I think that's what we measure our success on. So I think for an institute like ours and our partners, this is the way to go.
Merci beaucoup. Donc je retiens dans les clés succès, c'est déjà l'identification d'un intérêt commun pour tous les partenaires qui vont travailler sur le projet et l'accompagnement de jeunes chercheurs à travers des programmes de bourse et de financement. Donc je vais me retourner vers mon collègue Yap pour dire, alors à votre avis, quels sont les aspects qui sont les plus sous-évalués dans la collaboration, dans la recherche au niveau international, spécialement dans le contexte nord-sud ou bien les relations sud-sud, et qui nécessiterait qu'il y ait des discussions pour que ces types de réseaux aient un meilleur succès.
Merci beaucoup pour l'invitation et pour la question. En entrant dans la salle, il y a un poster qui met en avant les différents facteurs pour la recherche. Et le premier, c'est Equitable Partnership. On a également mentionné dans les valeurs principales du réseau, l'équité. Et je pense que l'équité aujourd'hui, c'est l'un des challenges majeurs pour nos réseaux. Si je demande à l'audience sur 10 articles qui sont publiés sur des recherches faites en Afrique, combien, d'après vous, ont un auteur africain qui soit premier ou dernier? Entre 1 et 10. Une réponse? Out of 10 articles published for research implement in Africa, according to you, how many of them will have an African first or last authors? 3. Okay, he's very right. So I had to speak in English. So, and that for us, it's a real challenge. It's a challenge. Why is it a challenge? Is because the first or the last author are actually the one who carry the responsibility of telling the story. So if 7 out of 10 articles have a non-African as first or last, while they are implementing Africa, there's a challenge. And I think that's really something that we need to address as African institution, but also as partners and even as ethical committee. We all submit our research to ethical committee. They also need to follow and ensure that at the end of the day, we have the adequate recognition. Why is it important? It is important because it shows that we have ownership. We have ownership of our research. We have ownership of our findings. When you have those results, then you will be the one to carry them on, to go and see the Ministry of Health, to go and see the community, and to ensure that there's the adequate research uptake. So, to you, it's really equity.
Yeah. Thank you. So we are extending the list of things that make the collaboration successful. So I'm going to switch for just one question in English, and I go back to French. So since my friend have influenced me. And so I have a question to Florence, since you have presented a lot of a lot of research you are running, and you have a huge capacity building program. So the question is, you are navigating on 16 countries in 3 continents. So how do you navigate the nuancing across the different continents and countries? How do you achieve this?
Merci, je vais continuer en français du coup. Donc oui, c'est vrai que le réseau Gabriel, il est présent sur 3 continents, 16 pays. Ces diversités géographiques peut être une richesse, une force, mais il y a beaucoup de challenges et il peut y avoir des difficultés. Alors pourquoi c'est une richesse? Parce que ça nous permet de mettre en place des études multicentriques. Par exemple, quand on a commencé le réseau, on a implémenté une étude sur la pneumonie chez les enfants hospitalisés de moins de 5 ans pour étudier l'étiologie, et cela a été réalisé avec 9 partenaires, et du coup ça nous a permis d'avoir des données sur l'étiologie de la pneumonie, de les comparer entre les différents pays. Ça permet aussi d'harmoniser les protocoles. Quand on met en place des études multicentriques, c'est vraiment important d'avoir la même méthodologie, d'avoir les mêmes... c'est pour ça qu'on développe les SOP, on les partage, on les harmonise pour pouvoir comparer les résultats. Ça permet également, en fait, les membres du réseau s'entraident. Et par exemple, dans le cadre d'un projet sur l'AMR, on a l'équipe brésilienne qui a accueilli des jeunes chercheurs de Madagascar et du Cameroun pour les former sur la métagénomique dans le cadre de leur projet. On a également des collaborations au sein du réseau. Certains membres, par exemple au Bangladesh, il y a 3 membres du réseau et il y a beaucoup de collaborations entre eux. Et par exemple, un membre a formé l'autre sur le séquençage. Donc on essaye vraiment à la fois d'avoir des interventions, des collaborations internationales, mais aussi essayer de renforcer au niveau régional. C'est pour ça qu'on structure aussi le réseau autour de thématiques, ce qui permet de fédérer. Et les difficultés, ça vient plus de la localisation. Nos membres ont des fois des difficultés dans l'accès des réactifs, par exemple. Des réactifs, ce qui est problématique et ce qui nécessite beaucoup d'efforts au niveau logistique. Et voilà.
La logistique. Oui, c'est vrai qu'au niveau international, la logistique est une question qui va être cruciale, surtout quand on doit déplacer du matériel ou installer des infrastructures pour les tests. Donc je reviens un peu à Dr Mariama. Donc vous avez parlé de ce que WANIDA a fait de bien pour vous et en quoi ça constitue une opportunité. Mais maintenant, quels sont les challenges que pose ce type de coopération et à votre avis, à quoi ressemble le futur de WANIDA?
Je pense que le challenge, c'est de pérenniser les acquis. C'est-à-dire d'essayer déjà de maintenir ce qui est là et d'aller à l'échelle, c'est-à-dire que d'avoir encore plus de collaboration dans des domaines variés et dans des thématiques diverses. Ça, je pense que c'est le challenge. Mais je pense que aussi, une fois que on a déjà commencé à collaborer, alors c'est facile de continuer cette collaboration et d'essayer d'associer d'autres acteurs, peut-être de même pays, pour pérenniser cette collaboration. Je pense également que c'est le lieu de dire qu'on peut mutualiser, c'est-à-dire qu'on va essayer, nous la Guinée par exemple, d'amener nos étudiants ici et qu'ici ils amènent les étudiants chez nous. Ça permet en fait de brasser et de permettre également d'avoir le réseau un peu plus large en partant également impliquer les étudiants au niveau du réseau. Et c'est le lieu de saluer en fait l'organisation de ce symposium, mais également de féliciter ceux qui ont eu le courage de participer au challenge Ma thèse en 180 secondes. Donc c'est pas facile, mais c'est également une manière de permettre aux doctorants de communiquer sur leurs thématiques en des termes plus simples, parce que quand on est doctorant, on a toujours les grosses thématiques dans la tête et c'est facile de communiquer dessus. Mais également de donner envie aux masters qui sont dans la salle d'aller vers le PhD, de dire que c'est pas compliqué, on peut y arriver. Donc je pense que le challenge c'est ça, c'est de permettre au réseau de rester soudé, mais également d'impliquer nos étudiants dans ce réseau-là, pour leur permettre de faciliter ou d'avoir une mobilité au niveau des différentes universités sous-régionaux.
Merci beaucoup. Je vais rebondir sur le mot implication, parce que CRIC implique plusieurs universités, 5 institutions africaines, et apparemment vous souhaiterez avoir plus de partenaires francophones. Et comme nous, on ne parle pas anglais, comme une blague, comment pensez-vous que ça va se faire, cet engagement d'institutions francophones?
I think that it's not a problem, to be honest. Between us, we'll understand, and if we really want to work together, we'll work together. Yes. We all learned French, mostly we've forgotten it, but we can always relearn it. And similarly, you've probably come across English, I'm sure you can relearn it. It will happen. It will absolutely happen. And our rationale for extending, we already have 3 or 4 Francophone fellows in our program who are just finishing, who are at Institut Pasteur or other places in Senegal and Côte d'Ivoire. So we know it works. We know that we can interact. And we've had a Beninoise who worked in the Gambia, who did perfectly fine, came to the CRIC, was perfectly fine. I believe if people are driven, they'll do it. But we have to, I would rather ignore the language barrier, to be honest. I worked in Germany a long time. I happily spoke any of the 3 languages that were around me with as far as I could go. I'm sure we'll manage. The CRIC has an understanding with the Pasteur Paris, and out of that came this discussion that perhaps we could extend our interests to the Pasteur Africa networks, and that's how this came about. And so I don't know how it'll go forward. We're going to talk about that tomorrow, a whole day of how we're going to find a way of interacting. But we already know that Francophone Fellows will join the next network. And I think as we go forward, we hope to extend things. I have to say, we cannot extend beyond what we can do. We have to accept we're a basic science institute with a lot of wonderful technologies that we can help people with. We can't do all the epidemiology, all the public health, much as we'd love to. We can, of course, contact people with our hospitals around us that collaborate with us to do that. But our value in this is to provide a kind of focal point for our partners to develop the fellows in their institutions. So while we may not increase massively, we will certainly incorporate new fellows from institutes outside our partners, and if people are willing to fund us, we'll extend. I mean, there's no point extending the partners if we only get £5 million and everybody gets 500,000. They can't do very much with it. So we've got to be realistic, but expansion is on the cards, and extension in terms of what we will do is also on the cards. We've listened to our fellows, they're all of one voice. 2 years is too short, and we need lots of technology, and we need to develop it in Africa. So those are the areas where we will seek to help them with a 4-year fellowship and more chance of developing their own technologies. And I think that's as much as we can conceivably do.
Très bien. Merci beaucoup pour cette réponse. Donc là, nous allons prendre peut-être quelques questions dans la salle avant de conclure. Il y a quelqu'un ici. Les micros. Ah, ok. Merci beaucoup. N'oubliez pas de vous présenter. Donc, on prendra les questions, ensuite on distribuera la parole pour les réponses.
Je suis Alabi Taoufik du Centre d'excellence africain sur le changement climatique, la biodiversité et l'agriculture durable en Côte d'Ivoire. Je vais rebondir sur la réaction de M. Boum. Je partage bien évidemment la question d'équité, mais la grosse problématique, c'est l'apportage de fonds. Voilà, quand on n'a pas la paternité des financements, on ne peut prétendre royalement à la propriété des résultats qui sont imbriqués. Donc, ce challenge-là, comment est-ce que vous l'envisagez en termes de solutions durables au niveau de l'Afrique? Voilà.
Oui, une autre question dans la salle? Ouais, David.
Thank you. I wanted to ask a quick question inspired by Prof. Yap's comments. When you talk about the lack of senior or first authorships on papers driven or actually derived from African science, what is the role, or how do we motivate the funders to make this an absolute requirement to have an equal seat at the table? Because so much of the bureaucracy that comes from the funders themselves starts from the premise of insisting on having a lead applicant from the US, from Europe. How do we change that and convince them that this is not the foundation for any form of equal partnership? You can't say that on one hand and do that on the other. How do we convince them of this? Because they like to say that they are equal, but then they demand the kind of the opposite. Where do we start?
Yeah, we want to answer after all the question is. So, maybe one last question. Yeah, before we give the floor to the panelists.
Yeah, thank you so much. I also wanted to talk about that issue. I mean, I've heard about many African scientists who participated in studies, and at the end of the day, when the paper is published, there's no African scientist in it. I think that is simply an issue of collaboration. There has to be discussion, within the scientific, the scientific group, in order to be able to determine right from the beginning of the project how are the papers going to be written, because it is an ethical matter, because you can't participate in the study, and then at the end of the day, when the study is published, your name is not there. That will discourage many people, in working, and or sometimes there are also students who need the papers more than the professors, so, and their names need to be in those papers so that they can be recognized, and that will encourage them and also help them to move up, with their careers. So I think that when there is a project and we want to embark on that project, it is important that discussions be made right from the beginning of the study to make sure that the African partners are not going to be left behind. Yeah. So I think that this is a culture that we have to adopt, even before the studies starts, because generally I've received many requests, from people from the US, from Europe, and I will tell them, how are we going to deal with publication? If you tell me no, it's going to be our partners, then I say, no, we can't work like that. And there is one issue that generally happens, is that many Africans are simply used as sample collectors. And we cannot continue like that. We just have to stop. We have to say that, no. If you want to work with our samples, you have to either take a student, somebody is going to go and work on those samples as part of, capacity development, or, you have to be able to do some of the studies here, but we have to be partners, we have to be equal partners, and we have to participate, all of us, in the publication. Even if sometimes maybe if their writing is not good, then, we can work together in order to be able to improve the quality of the paper, to make it high quality paper. Yeah, that was my contribution.
Ok, merci beaucoup. C'était en même temps une question et une contribution. Et Yap est très populaire. Il a touché apparemment un point sensible, donc je vais commencer par vous. Donc maintenant, si on peut avoir vraiment des réponses concises en maximum 2 minutes, parce qu'on a déjà dépassé le temps. Je sais que c'est un challenge, mais vous êtes des chercheurs, donc vous allez trouver la solution.
Merci bien. So first thing first, we need to admit that hard things are hard. Hard things are hard. It's really a challenge to decolonize global health. You've mentioned the issue of funding. The funding source should not impose those rules, and I agree with the last professor who was speaking. We need to agree as we start the study. Even when we say that, I recall I was in Uganda implementing a clinical trial on malaria. And we were working with some partners from MGH, Harvard University. And then we have to put the study on standby before starting, during 6 months. Because we were not agreeing on who will be the first and the last, prior to the study. So it's really hard, but we had, as you mentioned, to push. So the actual issue, it's not just us as African being first or last as a token. It's actually how do we invest in build the training capacity. We're discussing with, I think, Global Africa. WANIDA also has some training possibility. CRIC may also help. We need to train our young researchers, to train on how to write, because even when they have the data, are we all able to write those articles? And most of them in English. Most of the time, no. We also need to appreciate that. So it's critical to invest on those training and to look for the fund locally. It's interesting when you look at this bandroll. You see World Bank, AFD, ERD in here. Where's Africa CDC? Where's the Africa Development Bank? Where's Dangote? So we also need to go and find this local money that will give us more power on promoting our young researchers. Maybe I will stop from there.
Thank you. John?
I think David and this gentleman's point were absolutely right. And I think, in a way, I'm just going to blow our own trumpet again. I think the way we've done it is the CRIC PIs are not on these papers. Because they are from the African young scientist in the African institution. And I think that's how they took ownership. And I think it's taken ownership of it. And we should be telling funding agencies that there shouldn't just be one PI, to be honest. I've never understood that. I understand you might need one financial centre, but why do you only have to have one PI? Why can't we be telling wherever NIH or whatever, that it's not about PIs, it's about a real consortium and taking ownership of it. And I think we are starting to train our early career scientists into doing that and into applying for funding themselves, rather than relying on a European partner or an American partner to apply for the funding. There's no reason why somebody from here can't apply for Wellcome funding. They just got to be brave enough to do it and get the right support to write the grant, and that's what we should be doing.
Thank you. Before giving the floor to Florence, you have some example, but quickly, very quickly.
Thank you. Not actually example, but we wrote 2 articles. The first one is the dual equity checklist, which I invite you to go and see. It was published in PLOS Public Global Health, who gives some checklist on what you should look for before starting a study. And the other one is behind the scene that was published in the New England, highlighting the fact that when you look at the overhead, especially those provided by the NIH, if you are a US-based organization, you can go up to 50% overhead. But if you are a non-US based, the maximum you can get is 8%. So you can read and see how we can actually advocate to have towards more equity.
Thank you. And also we have Florence. Also, maybe you can answer to this question also.
Moi, je pense, je suis aussi tout à fait d'accord concernant le fait qu'il faut vraiment renforcer les capacités d'écriture scientifique, que ce soit pour les publications que pour la recherche de financement. Il y a quelques années, on avait essayé de lancer des ateliers de formation. On a dû en organiser 2 ou 3 avec donc, une personne qui était anglophone et qui s'adressait à des jeunes étudiants francophones, avec leurs données, et donc essayer à partir des données qu'ils avaient obtenues dans le cadre de leur recherche, de structurer un article, comment faire une introduction, comment faire matériel et méthode, etc. Et après de les suivre jusqu'à l'acceptation. Mais bon, on n'a pas eu de financement après pour continuer. Et je pense que ça, c'est une activité qui pourrait être inclue dans un module dans le cadre d'un programme de PhD au niveau universitaire. Parce que c'est vraiment la base de tout pour un chercheur. Rechercher des financements et publier ses résultats.
Je suis parfaitement d'accord avec ça, d'autant plus qu'aujourd'hui, dans beaucoup d'universités africaines francophones, on s'inspire du modèle anglophone. Parce que nous, nos thèses de doctorat n'avaient pas d'enseignement à proprement parler. C'est le doctorant et son directeur jusqu'à la fin de la thèse. Mais maintenant, on installe de plus en plus des modules de formation et les modules, pas forcément de rédaction de projet, de rédaction d'articles, mais de rédaction de projets, sont nécessaires même pour les enseignants-chercheurs, parce qu'on a beaucoup d'enseignants-chercheurs qui ont, pour les aspects scientifiques, on n'a pas à s'inquiéter. Mais quand il s'agit d'aller chercher des financements, ils ont presque peur de commencer à écrire. Donc je vais donner la parole peut-être à Mariam avant de clôturer, ou si tu veux ajouter quelque chose autour de l'équité ou la manière d'atteindre l'équité scientifique dans les collaborations dans les réseaux.
Je pense que ça, c'est un gros sujet dont on pourrait parler toute la journée. Mais bon, je vais essayer de faire court. Je pense, comme vous l'avez dit, tout à l'heure on en parlait, souvent les chercheurs, ils ont des idées, mais après coucher ces idées sur le papier, c'est difficile. Et argumenter ces idées pour pouvoir aller chercher des fonds, c'est aussi difficile. Donc, je pense que ça pourrait faire l'objet de formation et de renforcement de capacité au niveau des chercheurs pour les pousser à aller chercher des fonds. Mais j'adhère également au fait qu'il faut déjà voir au niveau local, est-ce qu'il y a des bailleurs de fonds. Parce que parfois, il y a des institutions qui ont des lignes et qui ne savent pas ce qu'il faut faire avec ces lignes. Mais il n'y a personne qui demande. Donc, quand on ne demande pas, on n'a pas. Donc, deux fois, il faut aller chercher au niveau des institutions, au niveau local. Parfois, il y a des choses à grappiller. Même dans les ministères au niveau local, parfois, il y a des choses qui dorment et qu'on peut aller chercher. Donc, je pense que si on est financé par des fonds au niveau national, au niveau régional, c'est plus facile de batailler pour le premier auteur. Voilà. Parce que là, on n'a pas à rendre compte à X ou Y, mais on a à rendre compte au niveau local, au niveau régional. Et c'est vrai que parfois, pour les chercheurs qui ont envie d'aller et qui ont besoin d'articles, ça pose problème. Ce n'est pas la compétence qui manque, mais parfois, c'est qu'on n'est pas dans le bon consortium et on a des difficultés pour publier.
Merci beaucoup, Mariama, pour cette brillante contribution. Donc, je vais essayer de résumer en 2 minutes tout ce qui s'est dit. Donc d'abord sur tout ce qui touche les bénéfices de ces réseaux-là Il y a le potentiel d'échange à l'échelle régionale et les collaborations inter- et pluridisciplinaires. Mais parmi les facteurs de succès, il y a l'identification de sujets et de projets d'intérêt commun, que tout le monde y voit. Ensuite, l'accompagnement et la formation des jeunes chercheurs à travers des programmes de fellowship. Ensuite, la pérennisation des activités ou des acquis par rapport à ce qu'on a déjà fait sur les réseaux. Et ensuite, essayer de voir comment on passe à l'échelle à ce niveau. Ensuite, il faudra gérer les différences culturelles qui sont en général facilement dépassables s'il y a la volonté de travailler ensemble. Et ensuite, s'il s'agit de l'équité scientifique, pour moi, soyons plus combatifs et proactifs. Et ensuite, essayons de faire du lobbying et de l'advocacy pour que ça marche. En tout cas, je vous remercie beaucoup d'avoir participé à ce panel et je remercie aussi WANIDA de me faire l'honneur de me confier le chairing de ce panel. Donc, je vous remercie le public d'avoir bien écouté et je remets la parole aux organisateurs. Merci.
Yeah, I know. Okay, so we will start the closing ceremony in a few minutes. Our guest of honor is arriving, so in 5 minutes. Thank you.
Time to wrap up.
Yeah.
We've been doing it, but we'll continue. No, it's African. We should have had. What about the postdoc? Do you have the address?
Yes, they have it loaded.
So they can pull it up.
Miriam, the adverts.
Advert for closing ceremony.
Hello. So I'm happy and sad at the same time. I'm happy that we've had a fantastic 3 days, absolutely beyond our expectations. And I'm sad that you guys are all going home. So how do we resolve this conundrum? Yeah, round table. It means that you come back again, right? You come again. So anyway, my friends, it's been a great pleasure hosting all of you and having such great, scientific discussion as well as networking over the last 3 days. We are so honored as WACBABE, as WANIDA, as University of Ghana, as Africa, to have had this meeting and hosted all of you and had so much productive discussions and had so much fun. So let's appreciate each and everybody. Before I say the thank yous, we have some business to do, which is to give awards. We always encourage young people to cooperate and collaborate, but we also encourage excellence and outstanding work. And so we like to recognize outstanding work. So one aspect of being a successful scientist is to be able to compete. Competition is not bad. Sometimes you win, sometimes you lose, but you learn through the process. And so we recognize the winners today, but we also appreciate the efforts of everybody who participated and put in an effort. So first, let's appreciate everybody who participated. Now, I hear that it was so competitive because everybody did such a great job for both the thesis, 3-minute thesis presentations, as well as the poster and oral and everything. Everybody did such a great job, so it was very competitive. So it was either we don't give awards at all, or we give a few. So Peter and the team will explain to us how they came by the award winners, and then we would present the awards. Thank you.
Thank you. Mampenda. So for the 3-minute thesis, what happened is we had 4 judges, and we were all very close. And we had 4 clear people who distinguished themselves. And we really, it was like a whisker between each of them. So we spent probably an hour, more than an hour, trying to, okay, not 3 hours, trying to figure out between them. And we came up with something that we hope will satisfy, well, it satisfied the jury somehow, right? Any? So for the 3-minute thesis, we were able to get 1 clear winner, 2 tied, second and third place. Yes. Oh, fourth, yes. I thought about it when I was, yes, fourth place. Should I announce?
Okay.
Okay. We'll let the chairman announce.
No. So,
I don't have the names.
Okay, so you give them to me in the order in which we present. So this for
No.
3 minutes.
Yes.
Yeah, we have 4.
Let's start from 4 to... Oh, 4.
All right. So I'm going to call people at random to help present. So Jack, please, can you help present the 3-minute thesis contest? So there are 4 winners. So we start with Julian Campbell. Wait. Next is Ali Badara Tuari. Oh, I forgot to read the prize. These are very handsome prizes, so please, excuse me, let me read the prize. This is a prize of 1,000 euros to participate in a scientific event. So 1,000 euros.
The first one was 500.
Yeah. Okay. So this is another 1,000 euros, and this is for the second prize now. This goes to Fatumatu Bintu Traore. And then the first among equals with a prize of 1,500 euros to participate in a scientific event and the production of a comic book about your research. So it's quite a nice prize. So we have Huefa Adeline Tatiana. All right. Dr. Jean Langon, can you make the next presentation for us? Thank you. Which one? This poster? There are 2?
No, poster 1 and then oral 1.
Okay. So you stand this way. Stand this way here so that. So this is best poster presentation. A prize value of 1,000 euros again to participate in a scientific event. So this is for travel. And the winner is Becky Tete. She wasn't around.
Gone for coffee.
Okay. Any friend of Becky here to take the honors? You don't look like Becky. What's that? Let me make the correction. All right. Then the best oral presentation, also 1,000 euros to participate in the scientific event. Issa Dieng. So we're done? Okay.
Go quickly.
Think we're done?
I like that one. I'm done now.
Yes, thank you very much, Jean. Thank you. All right, so business is done. It's time to say thanks. I want to start with our funders. So ACE Partner is funded by IRD and AFD, so we want to appreciate ACE Partner. And all this would not be possible without the ACE project itself. Okay, so the ACE Impact Project, which is the foundation of all this. And Nikes reminded me to not forget to mention the mechanism of funding of ACE. It's quite interesting. We often criticize our governments for not supporting science. But under the ACE, they were tricked into supporting science. So the way the ACE system works, the funding mechanism was that the World Bank asked for countries to express interest to participate. And signing up to participate meant that if any institution in your country won the grant, the World Bank will give the money to the institution and charge it to your national debt. You get the connection. I think it's a very clever way of getting African governments to fund science. They would never put $8 million into WACBIP if we didn't play this trick. They wouldn't put $8 million in each of those other centers and then put in another 6.4 million the second time if we didn't play this game. So to their credit, they allowed themselves to be tricked. So let's appreciate them. But I think it's a clever mechanism that the World Bank used. And I think other funders should think about that. When we think about co-funding, let's use this trick. Let's ask the countries to accept it as a loan. Okay? Most African countries, they don't really hesitate in taking loans because they know that we will pay in the future. They don't have to worry. So they are very happy to take a loan. Okay, so I think it's a great funding mechanism. So we thank the World Bank for a clever mechanism to get our governments to put some of our money into science without feeling it. All right, so for this conference in particular, we want to appreciate the scientific committee. So we had a scientific committee that, designed the program, called for abstracts, did the scientific review of the abstracts, and then, made everything happen. So I was part of that scientific committee, but I'll be honest that I did very little work on the committee. So let's appreciate those who actually did the work. Gregory was one of them. Nikes. Peter, where's Peter Kwashi? And then we had reps from each of the centers, right? I don't have the list here, but all those who supported from each of the centers to be part of this. But these were the core team who worked to, set the basis for this meeting scientifically. Then the logistics, which is even more difficult than the science. Putting a meeting together like this, the science is very little. You can do that from your bedroom, right? You can organize the science from the bedroom. The logistics is a nightmare. So we need to appreciate those who did the logistics as well. So let's first appreciate Olivia. Olivia, please stand up, yeah? So I'm the chairman of WANIDA, but Olivia is actually the operational chair. Because everything, she tells me what needs to be done, and I say, yes, madam, please, let's do it. Okay, so mine is just to approve. She comes up with all the ideas and does all the heavy lifting. I just approve and send emails. That's all. So massive thanks to Olivia for everything WANIDA. Then we had support from Sabrina's group with Marian and Zainab. So they also did a lot of heavy lifting. So please. Then we come home to the WAGBIB machine. Yeah? You know what the WAGBIB machine is? Yeah. The WAGBIB machine is the invisible hand that makes sure that your water is on the table, your rooms are there, you're picked up from the airport, everything. You don't have to think, you know. They think for you. That's what's called a WAGBIB machine, which we've built over the years to be able to deliver research projects and to deliver meetings and conferences and workshops. So let's give it up to the WAGBIB machine. These people, they make me famous because it took me a while to assemble the best people to do each particular job. But once I had them, then I went to sleep, okay? Actually, I went on to do other jobs so that they will do the work, okay? But these people are amazing people. They are some of the best people to do each of the jobs they are doing, so I really appreciate the effort that they put in. So, you know, Sika, you know, Andrew is our communications guy. Gloria is there, Felix, you know, everybody, the entire machine works very well. Thank you. And I want to thank all of you for making the time to come, spending 3 days of your time with us, and with Crick and Pastor continuing tomorrow with an extra day. It's been amazing. We're very grateful. We're very honored. And we hope that we'll see you again soon. Once you come into the WAGBIB family, you remain part of the WAGBIB family. Stay in touch with us. Check our website, opportunities for more engagement, workshops, conferences, you know, fellowships, you know. Right now we have a postdoc fellowship for the WWW consortium that David talked about. So check out the advert on our website and on our social media. If you fit the bill, please come and join us. We want the best scientists on the continent working here. So please apply if you can. And keep looking out for opportunities. And also, you are an advocate for us. You've come and seen what we're trying to do here as an African institution. We hope that you carry the message and tell everybody that there's something going on here and we need the support to continue going. So I thank you very much and I'll see you all soon. Good night.
Yes, so I think the meeting is over. It's time to network. Just have fun. There's coffee out there. So we have our coffee break and then that's it. So interact and please leave your headset. Oh, okay. That's the advert over there. So please take note of that and then, you know, you could apply. And kindly leave your headset on your tables or chairs for us to pick up. It is amazing having all of you around. So let's network, let's have the contact, and let's keep rolling. It is a pleasure to have you in Ghana for those who are, you know, the first time visiting Ghana. So that's it.
You can also announce. You can announce our social media platforms. We are. Yes. So I just want to add that we are present on all the various social media platforms. Just use the search word WAGBIB and you find us there. Thank you.
Thank you. So just a last reminding that we are working with a journal to have all your abstract be available, published online. So we will send a mail to all those who have presented to can check their abstract and online. Thank you.